Hear you.
Should we start now?
Hello, can you hear me?
Yeah, I can hear you. Should
Yes.
we start now?
Yes, let's start.
So can you just tell me when we go, if we're live now, or we go live and I'll start some of the opening remarks?
Professor Ian, we are live now and we are good to go. So
Perfect.
let's start. Yes.
So let me welcome everyone. It's lovely to welcome you to Healing Earth 4, which is the 4th edition of the flagship conference at the Centre of Climate Change and Sustainability, 3CS as we call it, at Ashoka University. I'm Ian Stewart and I co-direct the centre along with the director Gautam Menon, who's also with us here today. 3CS is embedded in Ashoka University's liberal education programme, which aims to enable critical thinking, complex problem solving, effective communication, interdisciplinary working and innovation for impact and change. And in 3CS, we assimilate those attributes in our mission of studying, communicating and mitigating the effects of future climate change on our society, health and environment. Climate change, global heating, the ecological breakdown is all redefining the way we see and relate to the world around us and increasingly reshaping the way that we live our everyday lives. Spilling out of nature is a series of pressing societal challenges: increased food and water security, large-scale displacements of populations, rising sea level, biodiversity loss, disease spillovers, increasing climate events. So, and in the face of all those coupled socio-ecological transformations, our current mode of living is increasingly becoming unstable and unsustainable. So the strapline in 3CS is that we're committed to awareness, research and advocacy on issues of climate change and sustainability. And in that sense, we harness Ashoka's wide and diverse and deep research and policy expertise, drawing from an intellectual community of scholars that show truly disciplinary academic engagement and blend the research interests of faculty members across 30 multiple departments. The result is corralled into a portfolio of thematic areas as diverse as climate change economics, environmental humanities, One Health, sustainable agriculture, ecosystems, climate and society, and sustainable communities. And as part of that, we also pay attention to engaging students with questions of sustainability, such as through the Live Green Campus initiative, using the Ashoka University campus to build a broader awareness and engagement on sustainability issues. A major focus is on communication, dissemination, dialogue, participation, partnerships, conveying the urgency of issues around climate change and sustainability through novel and unusual communications that provoke people to think differently about our rapidly changing world. One example is the 3CS Media Fellowship, which supports reportage on climate change in India to understand how it affects people's lives, and in particular the lives of those who live at the margins of society. Another example is this conference. It's part of our outreach portfolio. Today, we welcome the second of our two-day programme exploring frontier issues in climate change and sustainability. The first day yesterday addressed questions in urban ecology. Today's one is more broadly around climate change and health and ecology, and it blends two of 3CS's thematic specialism. The first is ecology, conservation and climate change, which is the group studying the nature of degraded ecosystems and how climate change impacts ecological aspects of our natural world. And the other is One Health, which places human health within the larger context of ecological health, whose contours will be determined by climate change, habitat changes, and biodiversity loss. Today's talks have been curated and coordinated by Ashoka colleagues Dr. Ghazala Shahabuddin and Dr. Amroz Khan. And now I'm going to stop talking and hand you over to Ghazala, who will kick off today's session, explain more about the theme and context, and talk you through our speakers. So over to you, Ghazala.
Thank you, Ian. Great to have you online today. Good evening to everyone who's joined and all our distinguished panelists. I work at the Environmental Studies Department at Ashoka and at ATRI, Bengaluru. On behalf of 3CS Ashoka University and my co-organizer, Dr. Imroz Khan, I would like to extend a warm welcome to our panelists, Dr. S.P. Singh, Dr. Sambuddha Mishra, Dr. Jessica Metcalf, and Dr. Meghna Krishnadas on day 2 of Healing Earth. There's no doubt that today climate change is one of the major anthropogenic influences on species, populations, and communities. It likely alters a range of ecological interactions and processes within ecosystems. New research within India and elsewhere now indicates a range of species losses, community restructuring, and weakening of ecosystem processes like pollination and water balance due to warming. Recent studies also suggest very close feedbacks between nature and human society as we witness the spread of infectious disease across the globe. On day 2 of Healing Earth, therefore, we decided to invite leading scientists who can give you a flavor for the trends and patterns being seen through recent climate change research. They will talk on a diverse range of issues, all the way from ocean acidification to the Himalayan tree line to forestry regeneration. In this context, I would like to introduce our very first speaker in this, Dr. S.P. Singh. Dr. S.P. Singh's, his video is already turned on. He is a leading expert in Himalayan forest ecology, and he's written some of the most radical, the most foundational papers on Himalayan ecology. He has been a professor and head of Department of Botany at Kumaon University in Nainital from 1987 to 2005. He was also the Vice Chancellor of H&B Garhwal University, Uttarakhand from 2005 to 2008. He's been associated with Himalayan ecological studies since the 1980s. The scales of his study range from a tree leaf to a complete forest ecosystem level analysis. He's written extensively on forest productivity, nutrient cycling, water relations, and sustainability initiatives more recently in Himalayan forests. He also studies lake and grassland ecosystems in the Himalayas. He is associated with numerous government institutions such as the Indian Council for Forestry Research and Education, Indian National Science Academy, and the Planning Commission of Uttarakhand. Today, we are very eager to hear him as he'll speak on the impact of climate in the Himalayas, which is still, despite its extreme importance for the Indian plains and for the Indian population, still very understudied. And he will refer particularly to the alpine tree line and how it's changing. Thank you for coming here, and over to you, Professor Singh.
Thank you so much for kind words, Ghazala. So should I start making presentation immediately?
Yes. Yes.
Well, I'm really thankful to you all for enabling me to share some of the things which I know about the Himalayan region in context of climate change. It's you know that there are many regions which are very vulnerable to climate change, and Himalaya is one of them. If mostly people talk about, say, coastal regions and Himalaya, as far as India is concerned, so my presentation would consist of three things. It will just give an idea of why Himalaya is important, how climate change is taking place, and its impact particularly in relation to tree line, which indicates the upper limit of altitude up to which trees can grow in natural conditions. So almost everybody knows about, say, that Himalaya is important, and you must have come across many papers on glacier melt. Glaciers are shrinking, or we may lose some of the glaciers very soon, or losing them. But there are many other aspects of climate change. The ecosystems are also responding, and they are being impacted because of climate change. So this presentation would be considering all those aspects, given an overview how ecosystems are responding, not only limiting to glacier melt. So we call Himalaya the water tower. The Chinese call it third pole because it has more snow and ice than any other part in the world outside the two poles. It has more species than say many. It is hottest spots of biodiversity. Endemism is very high. And you can just see that we don't know exactly how much species are there because 35 new species per year were discovered from 1998 onward. And it has forests up to 5-kilometer elevation. That's really something which makes Himalaya different. It has the highest tree line in the world that goes up to 4,900 meters. And the 5 kilometers of the vegetation in terms of height is something which is not seen in most of the mountains. Then it's 10 river basins originate. They are, say, where some 1.9 billion people live. And some of them are, say, in India, which are very important, like Ganges Plains and Brahmaputra, Indus. They are affected to varying degrees because of the climate change, and also because the amount of contribution of snow and ice as far as the river discharge is concerned varies. So overall assessment is that there are many data which keep on saying different things. But what we know is that the Himalaya has more snow and glaciers in western part than eastern part. And the warming rate is more in western part because of several factors, one of them being that the loss of glaciers and snow is leading to reduction in the albedo. And that may be one reason why the western Himalaya is warming at a higher rate than eastern Himalaya. But in pockets, you can find, say, temperature rise 2 to 3 times more than 1.5 degree, which is global average. You're going to say so. It varies a lot, but overall trend is that the temperature rise has been much more than the global average. Some other indications are that if you look at, say, days, the number of days which are warmer than they used to be normal, and nights, all of them are increasing, and the number of days which are cooler than normal is declining. And the difference is much more in between day and night. I mean, so night is warming much more than day, and winter is warming much more than, say, other years of the seasons. So we are, in general, you can briefly say that nights are warming faster than, say, days, and the winters more than the other seasons, which have a lot of implications in different ways. Overall, some people say that some glaciers are stable or they are swelling, but overall scenario is that 75% of glaciers are shrinking. As far as Karakoram is concerned, which is the area where we say that some of the glaciers are stable or they are expanding. But recent research show that things are no more that rosy. Even there, the problems have started, and that the idea of stability of those glaciers is questionable now. Then one of the factors which what they do is that it leads to formation of glacial lakes and then outburst. That's why people say that, especially the organizations like ECMOD, that there should be much more collaboration among the countries than it has been if we want to deal with the problems of glacier lakes, outburst, or so on. Because a glacial lake of one country may be affecting the people of other countries. So we need a lot of cooperation, even if, say, a level of certain understanding should be there. Unfortunately, Himalaya has many international boundaries, and the things are not as good as they should have been. But anyway, to address those common problems, there's a need to have a more cooperation, and that is what people are saying. So the next point is that how glaciers are affecting us, or how they're going to affect us. The predictions say that Ganges Plain, I'm saying the Ganges are not going to lose much water discharge because it is more monsoonal than, say, glacier fed. But as far as Indus is concerned, things are dire, and Indus may lose something substantially because it is fed. Its discharge is about 60% from glaciers and snow. Brahmaputra is somewhat midway, but overall scenario is that during summers things would be when water is most required. things would be worse, and we need to really, we can see that some of the changes already taking place. One of the consequences of the glacier, say, as far as ecosystem is concerned, is that because of pre-monsoon is getting intensified as far as drought is concerned. You have a monsoon, but nobody knows, people hardly talk about pre-monsoon when when the drought is severest. And you can see that Himalaya throughout the Ghazala knows it, that she must be familiar with the fire which normally occurs in pine forest during pre-monsoon time. So there's a need to really predict how pre-monsoon is going to behave in a given year. You can see some of the data which shows that when there's a certain amount of pre-monsoon showers can stop say fires, but when they're not there, the fire intensity becomes higher. So we need to have a better understanding of pre-monsoon drought to understand or to prepare as far as drought is as far as forest fire during pre-monsoon is concerned. Then there are forest ecosystems which are more vulnerable. So there's pine is one of them. So there's a relationship between drought, the time of litter fall, and the pine forest ecosystems. So when these three combine, then you have a more fires than in other years. So some modeling and some understanding of these data, this kind of information is required. So we need to have not only better prediction of monsoon, but also we need to have a better prediction of pre-monsoon drought and its intensification, because fire is a serious issue also from another point of view. It emits a lot of black carbon, and black carbon is one of the factors which is causing glaciers to melt. So fire can't be taken simply as a fire factor, but also it's the pollution or impact on the glacier melt should also be seen. Then we have monsoon predictability, and monsoon may change as far as time is concerned, and also because of warming. Many species may start maturing seeds earlier. Some evidences are already there, especially oaks and many, sals, Shoya robusta, which are very important species of this region. They used to have a seed fall during just beginning of monsoon. And there was a synchronization between seed fall and monsoon. And they used to have a very short seed viability. Such species are going to be in problem because they may mature earlier than monsoon arrival, or monsoon may also vary because of climate change. Already some symptoms are there, some indications are there that sal regeneration is a problem, or maybe oaks are in problem. Then we have, some species where seed fall takes place during winter, and they lie on the ground throughout the winter. Now, because of winter warming, which is more than normal warming, these seeds are getting desiccated before they can germinate. Means by the time monsoon arrives, they're already desiccated. And one of them is the barge oak that is called Quercus leucotrichophora. In this respect, the other species, which is Pinus roxburghia, which competes with the barge oak, and much of the region of lower elevations where humans live is dominated by these two species. So pine is favored by fire, or pine leads to more fire, as well as it is favored by fire. Now, it is maybe favored also because if pre-monsoon becomes, or say winters become more drier, because this species can have a seed dormancy, and it can really manage to survive even if warming takes place. The other things are that there are certain species which occur only near the summits, mountain summits, and their patches may be 70 to 60, 70 kilometer distant. One of them is a very important species called Kharsu oak, Quercus semicaudifolia, which occurs in the close to tree line, and in a fashion like islands, which are separated. Now they may not survive because very little space is left, and they're very close to summits, and such species are also in problem. they say that this Semicarfolia may be one of the major species to be affected by climate change the earliest. Now then, we have problems also with, say, crops also, say like apple, which required a certain level of chilling, something like about 1200 to 1500 hours. And now such temperatures not available in lower elevations where apple belts are there. So you have a problem in Kullu and Shimla valleys where temperatures are warmer and chilling is not possible. So one has to shift to higher elevations, or one should go for new varieties which might have different kinds of problems. So the idea is that we need a lot of adaptation research is required to grow crops which require less chilling. Some researchers indicate that the apples with the 400 to 500 hours of chilling they have been developed, but we don't know about their, say, responses, and the experience is still quite new. Now, coming to this tree line issue, tree line means there is the altitude above which a species might not grow as a tree. The point is that, as we climb, there's a reduction in temperature that's called temperature lapse rate. The rate normally people used to assume that there is certain standard. No, our research showed that no, it's not. The temperature variations from one place to other, as far as lapse rate is concerned, is considerable, and it varies also seasonally. So things are more complex. Coming to this point, tree line means a place above which, or altitude above which, trees fail to grow and that temperature is something about something around 6.7 degrees Celsius during growing period, which should not be less than 94 or 95 days, something like that. So that is the condition which is required for trees to grow, and trees means, say, upright individuals which are woody. So as we go high, the trees become sparser. Then first there is the timberline, we call it, where we think that the canopy should be about 30%, above which then trees become more sparser. Grasses come, bushes come, like rhododendrons, something like that. And then it is all alpine meadows. Now the situation is that this tree line is expected to, say, shift upward because of climate change and intrude upon the alpine meadows where so many precious species from medicine point of view live. So I will come to this point later on. The glaciers, I just want to show that the tree line means what it is really in picture. It can be quite abrupt, the lower one showing, but it is diffuse type, means as you go up, the trees become sparser gradually, and then a situation comes when there is no tree. And then there can be some like finger-like diffuse tree lines, which is from Kashmir. You can see that because of, say, avalanche and shoots or others, because this cutting it causes, there are areas where you may not have tree lines at all. Trees can't grow because of just a minute. Sorry. So you have a situation in which the furrows have a lot of snow, so trees may not grow there, but ridges where the snow doesn't stay longer, they have trees. Now, because of climate change, situation may be just reverse. When the snow is gone, then it's quite possible that the furrows may have trees, not the ridges. So such situations, some changes like that are being seen. Now, the tree line, say in Himalaya, as I said, that it is the highest in the world. It goes up to 4,900. And there's a species called Juniper tibetica, which grows in the Tibetan plateau, which is highest in the northern hemisphere. But you can find tree lines elevation changing from one aspect to another. You know that the south aspect is warmer than, say, north aspect. So tree line can be higher in north aspect, south aspect. And then the species composition also varies. The tree line is consists of so many species, various species. And that species may vary also because of aspects and other factors. Now you can see here that we have on one fir and birch. Birch means Betula utalis and fir is Abies pindro or Abies spectabilis. The other side we have say fir with the pine on the drier side, means on the southern slope, and on the northern slope we have a birch along with the say fir. So even at the same place, the species composition can vary because of aspect. And also, I just draw your attention to the point that the Himalaya has two types of monsoonal systems. One is they call it the outer ranges, which receive most of the monsoon. Then there are areas which are shielded from monsoon, and they are severe rain shadow areas. So tree lines in rain shadow areas are higher than say outer ranges, and overall variations in tree line elevation in Himalaya can be up to about 1,700 to 1,800 meters. Means you can find tree line right from 3,000 meters to 4,900 meters, something like that. So Himalaya varies a lot as far as tree line elevation is concerned. And the patterns are decided by aspects, by whether you receive monsoon or not, or it's a drier shadow, rain shadow areas, and so on. So Himalayan tree line is interesting because of the elevation variations. Also because it has a very rich species composition. Globally, what we have estimated that not more than 150 or 60 tree species form tree lines in the world, out of which 58 occur in Himalaya, which is quite rich. I would say there are several species which occur in tree lines, and then beyond tree line, you have alpine meadows, which are in other form. So I was just drawing your attention to another point, that all these years people used to have a certain standard called temperature lapse rate. Means with a given elevation range change, the temperature would decline at a certain rate. That comes to about, say, but in Himalaya, what we are realizing that in Himalaya it varies a lot from one place to other. Sikkim can be very low, means say the temperature lapse rate, with declining temperature will be much slower than in Uttarakhand, and in Kashmir, which is more arid, it's much more. So the elevation, the changes in vegetation is also because of the difference in lapse rate. And this lapse rate also varies seasonally. Implications of these seasonal changes in lapse rate, for example, you are seeing in December, it's almost no temperature decline with the altitude, partly because the plains are getting say more say polluted, you can find more aerosols. And in the higher elevations, you can find that things are more sunny. And then also the glacier melt is taking place. So it becomes very dark vegetation during December, because snowfall is delayed now. Earlier, snowfall used to take place in December. Now it is delayed, and mostly it takes place in January, February, or something like that. So albedo in December is almost lowest. So it means what we call it elevation-dependent warming is taking place. Though globally, if you do global analysis, things are unclear. Locally, such phenomenon is taking place that certain portion becomes warmer because snow is not there. Vegetation plants can't grow during winters, and the whole area becomes much more darker than it used to be. So elevation-dependent warming is one of the factors why temperature lapse rate is declining in Himalaya. Means if this process continues, so vegetation ranges, species changes would be much more difficult to predict in view of these changes. So we are not sure about, say, what is the extent of elevation-dependent warming. but means warming more in higher elevation than lower elevations, or warming more in the mountains than in plains. But in Himalaya, there's some evidences are there. Then other thing is that most attractive question is that how tree lines are they moving up? Globally, global analysis showed that about 52% in instances, tree lines have been found to go up 48 knot. So they are in Himalaya. Our research shows that Subrat Sharma and his group had done used remote sensing methods, and they did it in Sikkim, Uttarakhand, Himachal, and found that say most of the tree lines are not going up; they are stationary. It means the trees are finding difficult to really go up because of several factors. So they have to, the tree line species are going to experience much higher temperature than they used to be. It's not going to be average temperature of 6.7, but it can be 8, 9, 10, 11. And already in some places, we found that temperatures are close to 10 degrees Celsius at tree line. Means more evapotranspiration loss, and it may lead to more mortalities in future. These things need to be really investigated. So one of the reasons why tree lines are not going up is that Himalayan tree line was already very high. Himalaya, because of the mass effect, they call it, is already quite warm before the climate change occurred. So the vegetation went up to 5 meter height as the tree line was concerned. So there's very little space left to say from that place to summits of the mountains. Then grazing was another factor. In fact, some scientists say that maybe the 50% of the tree lines which are going up, maybe because of the land use change, means grazing has declined in recent years. Because of the cultural practices, people are no more interested in grazing animals or having animals in those areas. So a new point comes that we might need grazing to really maintain our alpine meadows. Otherwise, say our tree line may intrude upon glaciers. These are, say, alpine meadows, and you may not have many of those species which occur in alpine meadows. The other is, say, terrain is difficult, and another factor which is coming up is as I said, pre-monsoon drought is getting intensified. Means the temperature rise, say, is one factor. Temperature normally should favor species to grow in tree line because there is more water available. There would be, say, better conditions for photosynthesis. But the drought may get intensified. So studies which are based on tree rings, they indicate that even in the past, wherever the drought was severe, the tree rings were very thin. So the growth was much more truncated than it used to be normal years. So it's quite possible that the glacier, the tree line which should have been favored by warming is not really, is maybe disfavored by the increased intensity of drought. So the tree line analysis shows that most of the tree lines are in Himalaya are not rising because of the intensification of drought during the pre-monsoon. So we need to really, the lesson is that we need not only understand the relationship of temperature and growth, but also how temperature is affecting the monsoon and the drought. We could study three, but there's the need to have, say, more sites to be studied, because remote sensing may not be completely reliable. You need a lot of ground truthing. Whatever data we have at present shows that Himalayan tree lines are largely stationary. And its implications, we don't know. These are some of the methods which people are using. The tree line width is being used to assess whether drought can be a factor. Past records show that such things are quite possible. Then this is another from Kashmir. You can see that how this is a very inhospitable environment where a process called facilitation by another species is important. And in this case, you are seeing that the juniper mat is supporting the growth of silver fir. Means the number of seedlings in juniper fir were more than outside juniper fir of Abies spindle. So the lesson is that we need to really take care of those other species which look very unimportant, but they play a role of facilitating role as far as species survival is concerned. That kind of knowledge and more experiments are required to understand how one species growth is facilitated by another growth and how we need to really manage things. The another interesting feature is the adaptation of species to tree line. What we found that there are two firs in Himalaya, especially in Western Himalaya. One is called Abies spindle, the other is called Abies spectabilis. So Abies spectabilis is a tree line fir, and Abies spindle is say it occurs in lower elevation. The differences in their morphology shows that how species adapt to tree lines. You can see that the CBH means the girth of the both species same almost, but the height of tree line species is lower. So there is a kind of stunting takes place. Means the girth is not affected, but the height is affected because the aerial conditions are very different than the ground conditions. So you can find some changes like that. And then you can see that the needle length becomes shorter. Other interesting point is that the number of leaves get, say, much more, means the leaves become much more denser. On the right side, they are much more denser than they used to be. So because of that, they are able to really, say, survive in those conditions. The details of these physiological factors, say, modifications, we need to understand. Other thing is that what is going to happen because glaciers are, or say, snow is going to deplete. Initially, we found that both species density of herbaceous plants and the species richness, they increased because of early glacier melt. But what will happen when glaciers, early snow melt, but we don't know what will happen when there is no snow at all. And so it is the question of only delay. Delay shows that there will be more growth of the herbaceous plants or maybe also woody plants. So overall, the dynamics of species, how they're going to change in future, needs to be monitored, and this calls for long-term studies. Otherwise, a simple 1 or 2, 3 years sampling may not be enough to really understand the processes. Some other interesting aspects which are occurring is one of them is the Changthang alpine meadows, the grasslands in Leh, where there are Pashmina goats. Now people realize that Pashmina goat used to have a low say very thin hair, largely because of low temperature, and that made them very especially warm. Now those hair, they're getting thicker and they will not be as warm they used to be. So the details of the implications of climate change can be there a lot. And then glaciers are getting, snow is getting melted earlier, timings have become different, and people are finding it difficult to really manage those herds of Pashmina goat. Now, some people say that there are many people abandoning this idea, this grazing practices, shifting to the cities. And there's a concern about they say that this is one of the cultural heritages of the entire humanity, where something like Pashmina goats, should it be maintained or not should it be maintained, and how it should be done in the modern times when people really, urbanization is very rapid, tourism is very lucrative as far as Ladakh is concerned. So there are many forces which are modern, which are affecting in partnership with the climate change. And the things are adaptations and factors, they become very important as far as climate change is concerned. There's another angle that Himalayan forests played a very important role in sequestering carbon. They still have a lot of carbon in their body. Now, the question of this, I call it carbon justice, that the women or men who used to collect fodder by bit in, say, headloads, they never cut the whole tree. Should it be considered a kind of, have they not played a role in slowing down the climate change? I think so, that by their consumption level means emission level of fossil fuel carbon was so low, almost negligible. So mountains in general have played a very important role. In a way, they've already played a role of slowing down the climate change by keeping their consumption of fossil fuels much lower than others, and also not cutting the whole tree or deforesting at a large scale. As you can say, nowadays we see that when there is road formation, construction, the widening road itself may cut, say, thousands of trees. Those villagers might have taken some 100 years to cut those number of trees. So the point is that there are 2 points as far as carbon justice is concerned. One is that these people don't know that they have provided some service to the humanity. We never told them that you have done something like this. So my submission is that those who had a very little fossil fuel consumption or emitted their contribution to in terms of emission was much low, they're going to suffer much more because of climate change, maybe water, maybe glacier melt, maybe landslides, and so on. So should there not be a sense of justice and provide them some kind of, facilities so that they can adapt better than normally they are? So global average is about, say, emissions. I want to draw your attention, is about 4.8 ton. So data differ a lot from one place to other. But just see Nepal, or say, it had only 0.5 ton per person per year emission from fossil fuel. Entire Nepal I'm talking about. There are countries like, say, Afghanistan, which are still lower. So the point is that, is there not need that those who hardly emitted fossil fuel carbon, they conserve forests to some extent, they collected only, say, leaves or twigs, or they didn't cut the whole tree. Should those services be acknowledged or not? This is another angle as far as Himalaya is concerned. So in brief, this presentation, I thought that I should draw your attention to 2 things. One is the glacier melt is a serious issue. But there are other points which are ecosystems are also changing. We need to have a better monitoring or understanding of these ecosystems. We don't know how many species are going to be collected at one place. There is a kind of, some people saying that the studies in Europe showed that the near summits, there is a kind of jamming of the species, means more species are getting collected near the summit. And then in the second phase, they may, some of them may become extinct because not all species can be jammed at one place. How these things need to be really assisted means should there be a kind of assisted regeneration or assisted transfer of one species to another for different places so that the species extinction is stopped. So we need to really have a better long-term studies if we think that we can address some of these Himalayan situations. So my appeal is that Himalaya provides a lot of ecosystems, a lot of systems, in fact, where you can do research. And those who live or those who don't live in Himalaya, their services are needed to do more research. The data is still our paltry, and we are not able to really decide most of the times. Debates continue simply because we don't have conclusive data. As far as climate change in Himalaya is concerned, Himalaya is a very difficult area. The glaciers or, say, tree lines are very remote, at remote locations. So we need more young people to really be involved to understand these processes of climate change. So I'm again thankful to your university, which which is, I've been hearing about this university and the kind of say programs you have that enabled me to share some of the experiences of my research as far as Himalaya is concerned. Thank you, Dr. Ghazal, again.
Thank you very much, Professor Singh. That was really fascinating. It just goes to show that things are going to be far more complex to predict. It's not easy to generalize about movement of species uphill that we commonly, talk about and tell our students. So there's definitely a need for more field work, to actually examine these things on the ground, and we don't have enough of that. I'm sure that people have a lot of questions to Dr. Singh. If you can hold those till we finish the second presentation. And now I would like to introduce Dr. Sambuddha Mishra. We're really grateful to you for being able to join us today and talk about a really exciting topic. And Dr. Sambuddha Mishra has earned his doctorate in oceanography in 2010 from Florida State University and the National High Magnetic Field Laboratory. He's currently working at the Center for Earth Sciences at the Indian Institute of Sciences in Bengaluru. He is basically a chemical oceanographer with expertise in low temperature isotope geochemistry. His primary research interest is in the reconstruction of past changes in seawater and chemistry and climate that it can tell us. Some of the problems that he has been working on include reconstruction of past changes in atmospheric carbon dioxide concentration, role of reverse weathering in modulating seawater chemistry, as well as the transport of metals and metalloids in large river systems. This evening, he'll speak on ocean acidification and its effects on corals. Thank you, Dr. Mishra, for being here. Over to you.
Thank you for having me. Am I audible?
Yes.
And I hope the presentation is also visible. Presentation is perfect. Go for the laser point. Perfect. So, what I'm going to talk to you guys today about is how corals act as a faithful archive of past changes in seawater pH, what we call ocean acidification. And we can't call it ocean acidification per se, which is caused by humans, because naturally carbon dioxide levels of the planet have gone up and down over the course of the history of the planet. So corals act as a very faithful archive for it. Now, we would think the corals are extremely vulnerable to these ocean acidification. So my lecture is going to focus on first establishing that why corals are vulnerable to ocean acidification, then demonstrating that they are quite robust against ocean acidification, but they are suffering greatly through global warming. So let's proceed. Now, coral reefs are declining, and it's quite visual. You don't need to be a rocket scientist to observe their degradation. This is a series of pictures from the Great Barrier Reef taken between December 2014 and December 2015. And you can see that they were healthy, then they were getting bleached, and they're all dead. Now, remember, corals are a combination of a calcium carbonate structure with symbionts sitting on top of them. Now, when we think of corals, we look at structures like this. This is a coral which is getting slightly bleached, but normally corals will be very colorful because they have these symbionts, zooxanthellae, living with them, supplying them with food, and corals are providing them with a shelter or a structure on which they can grow. So it's a combination of the two. Now, corals, when a common man looks at a coral, they see these colorful zooxanthellae. When I, as a chemist, look at corals, I look at them as a calcium carbonate structure, and specifically aragonite. So when we form calcium carbonate, we can form two different species, calcite and aragonite. Your chalk is calcite, the chalk you write on a board with. And aragonite are corals, which are, as we know, slightly more prone to dissolution if there is an increase in acidity. You don't have to get to the details of this diagram. What it shows that when these calcification or the biogenic calcite formation happens, the material sinks to the bottom of the ocean. And as it sinks to the bottom of the ocean, it passes a horizon which is called ACD. And the ACD is much shallower than the CCD. ACD stands for aragonite compensation depth, and CCD stands for carbonate compensation depth. Carbonate means calcite compensation depth. So aragonites dissolve way faster than calcite. That's because the solubility product of aragonite is much lower than the calcite. Now, biology has been forming these biominerals or calcium carbonate for a very, very long time. Since before the evolution of multicellular organisms, there has been formation of biogenic calcium carbonate. There are two ways of forming this calcium carbonate. One is by bacteria, where it simply pumps a lot of calcium and carbonate ion into its surroundings and takes out the proton from it, because we know if we add acid to any system, things will dissolve. Now, if you remove that acid, things will precipitate. So they can form these extracellular calcium carbonate. Now, the same idea was implemented by other single cellular organisms like foraminifera, who form these really marvelous calcium carbonate structures to host their single cell protease, so that the little cell that they have has to be protected from its environment, and they form these differently shaped, marvelous looking calcium carbonate. And why are they important? Because these calcium carbonates were one of the first indicators of ocean acidification. Now, we have been harping on ocean acidification for a long time. It's supposed to be the twin evil of global warming. Now, how do we know ocean acidification is happening? Here we have two panels of the same foraminifera species. The bottom one was collected by the HMS Challenger expedition between 1872 and 1878. This particular specimen was collected off the coast of Peru in the year 1877. And the Tara Ocean mission in the year 2009 to 2013, which ran through these years, collected the same species from the same exact location in 2011. So 1877 versus 2011. And what we are looking at is scanning electron images of the thickness of these shells. And it's quite obvious the Ruber shells have undergone extreme thinning because of ocean acidification over the last 100, 150 years. So ocean acidification is real, and how are the corals coping with it? Now, what is ocean acidification? Ocean acidification can simply be put as invasion of carbon dioxide in seawater. Now, naturally, there is a lot of carbon dioxide in the atmosphere. There's a lot of carbon dioxide in the surface ocean, but there is a lot more carbon dioxide in the deep ocean. So there is a constant exchange of carbon dioxide between atmosphere and the surface ocean. We're going to ignore the soil part now, but keep in mind one thing. As a layman, one would think that the soil or the biosystem above soil carries a lot of carbon stored in it, and we are looking at petagrams of carbon, gigatons. So the plant biomass contains about 550 petagrams, and the soil contains about 2,300 petagrams. And as you look into the deep ocean, it contains about 37,000 petagrams of carbon, which is way larger than any of these reservoirs. So oceans have an unusual property of absorbing CO2. And why does that property exist? Because CO2 is a reactive gas. The moment it comes in contact with water, it forms carbonic acid, and the carbonic acid will dissolve into bicarbonate ion. So if I dissolve carbon dioxide in seawater, and I have some way of consuming this H+ ion, which actually causes acidity, then all the equilibrium will move to the right of this equation, which means the ocean will keep absorbing more and more CO2. And in doing so, the H+ concentration in the ocean will go up. That means oceans will become acidic in nature. And the evidence has been instrumentally proven. This is a series of carbon dioxide concentration data from Hawaii, the station Mauna Loa. And the oceanographic data is from the station Aloha. On the left-hand y-axis, we have CO2 concentration going from 280 to 430 ppm from year 1958 to 2024. Keep in mind, this is an instrumental record. This is not reconstructed. And you can observe the seesaw pattern of the CO2 and the continuous rise in the CO2 concentration. On the right-hand y-axis, we can observe the increase in the sea surface carbon dioxide concentration and decrease in the sea surface pH. So CO2 is invading surface ocean and causing a pH drop. Now, oceans and atmosphere interact in a very dynamic way. It will be foolish of us to think that as CO2 concentration is going up, there is a constant flux of CO2 from atmosphere to the ocean, but that is not the case. In this diagram, anything which falls within the green and yellow region is a place which is neutral, where we have no net exchange of CO2 between the ocean and the atmosphere. Anything which is a lighter color, which is towards the violet, we have a strong invasion of CO2 from the atmosphere to the ocean, because these are high latitude places, cooler temperature, more gases dissolve. If you look at a real life experiment, if you have a bottle of Coca-Cola, you have left it out in the open versus it's in the fridge. When you open the bottle which has been left out at a warmer temperature, there are more gases fizzed out of it, simply because at a lower temperature, gases are more soluble in a fluid phase. Now, these peculiar red areas, which we observe in equatorial oceans, especially in the equatorial Pacific by the coast of Peru, this is what we call the ENSO El Nino situation, and Arabian Sea are places where waters from the deep ocean comes to the surface by process what we call upwelling. So it brings a lot of CO2 to the atmosphere and then degasses from the ocean to the atmosphere. It also triggers a lot of biological productivity. And we know for sure that corals grow in these regions. In regions where we upwell very low pH, corrosive waters, corals can grow. Now, that means corals have an ace up their sleeve. What is it? Now, what do we expect in terms of ocean acidification for the future? This is what the globe used to look like in 1850. Why 1850? Because we haven't perturbed atmospheric CO2 concentration as greatly by 1850 as we have now. As we call it, the pre-industrial value. And we expect the ocean pH to drop around 7.8, 7.9 by the year 2100. Now, these are the different scenarios in which the ocean pH is supposed to drop. But one peculiar thing is that the CO2 in seawater is not a linear system. Now, what we have here is pre-industrial CO2 concentration, 280, double that, 560, and triple that amount. Now, if you look at the different species formed at that particular level of carbon dioxide in the atmosphere, they are not scaling up linearly. The bicarbonate ion is increasing. The carbonate ion is actually dropping. And remember, calcium carbonate is calcium and carbonate ion. If I drop one of the constituents of calcium carbonate, the solubility of calcium carbonate will go up. And that is exactly what we observe with invasion of carbon dioxide in seawater. So if we don't reduce CO2 emissions in the modern world, this is what we will get. So we will have an ocean which is sitting around pH 7.6 to between 7.9. Can the corals live? The clue to that lies in the past variations in carbon dioxide. If we look at the Antarctic ice core records, these are the air bubbles trapped in Antarctic ice cores, and measure CO2 concentration in it, what we observe that the CO2 concentration usually varied between 300 ppm to 180 ppm. And in the modern day, we have caused an alarming rise in CO2 concentration. Everything should die off. No. If we go back in the past, this graph shows CO2 concentrations over millions of years. We are going from modern day up to KT boundary when the dinosaurs died away. And you can see the CO2 concentrations are way, way above the 280 ppm threshold we had put for the last 1 million years. Do we find corals from these ages? Yes, we do. That means corals, which are aragonite secreting organisms, which are vulnerable to seawater acidification, have survived very high CO2 worlds. And how did they manage that? And what I'm going to do that is introduce you to the dependence of the proxy that we use to do seawater pH reconstruction and understand how biominerals are formed. Now, going back to the CO2 dissolves, dissociates into carbonic acid, bicarbonate ion, and carbonate ion. And if you plot it on a pH axis, this is what it would look like. You don't have to get all of it, but essentially, between the pH of seawater, which is between 7 and 9, these are the species that dominate. Bicarbonate dominates, there's a bit of carbonate ion. Now, seawater has a lot of dissolved salts in it, which is around about a kilo of seawater will give you around 965.5 grams of water. The rest of it is salt. And among the salt, sodium and chlorine dominates. Then there is sulfate, magnesium, bicarbonate, calcium, potassium. And there is this other component. And within that other component sits boron, which is a minor component of seawater. Now, remember the carbon dioxide speciation with pH? Boron, luckily for us, also speciates with pH. So if the calcium carbonate, which is being secreted by a biological organism, takes up boron into its lattice structure and takes up only one of the species, we now have a way of reconstructing seawater pH of the past. And that is exactly what happens. Now, the boron in seawater speciates between boric acid and borate ion. As expected, any acid will break down into its conjugate base, and which the borate ion is the conjugate base, and the conjugate base is taken up by biogenic calcite. So it faithfully records the seawater pH as it grows in an ambient environment. But remember the constituents of these salts in seawater, it's dominated by sodium, sulfate, magnesium, and chlorine. However, when biogenic calcite is formed from seawater, it is very different from what inorganic calcite constituents of. When inorganic calcite is formed from seawater, it has a lot of impurities in it, and that impurity is dominated by magnesium. It is expected. Magnesium and calcium fall on the group 2. They are both alkaline earth elements, so they have very similar chemistry. But when biogenic calcite is formed, you can see magnesium is all but gone. But the sodium and the sulfur levels are very similar to inorganic calcite. Chlorine is not present in biogenic calcite. How does that happen? Now, impurities in biogenic calcite are not homogeneously distributed, which means the organism itself, even if it's a single cellular organism, is controlling the microenvironment in which it is precipitating this calcium carbonate. There has to be some sort of ion movement from the surrounding seawater into that microenvironment and some sort of pumping out of ion from there. Why do we get rid of magnesium? Not we, the forams or the corals get rid of magnesium because it's a strong inhibitor for calcium carbonate precipitation. Now, there are two pathways by which these organisms can form their calcium carbonate. One is they take up the calcium and the carbonate ion from the surrounding to itself, or they take up a gulp of seawater and exclude the magnesium, exclude the other single valent cations. Not only that, the forams or the biogenic calcites, the corals, they are very clever. What they do is they upregulate their pH in the growth region. So this is a biogenic carbonate forming. The chamber is growing, and you can see the pH in the pH scale. The growth chamber has a pH of 9. Ambient seawater is supposed to have a pH of 8. So it is upregulating the pH. And the corollary evidence that it's upregulating the pH. How can I upregulate my pH? I have to get rid of proton. And the corollary evidence comes from the drop in pH in its surrounding environment because it's pumping the proton out. So now we have a template to demonstrate why corals manage to survive ocean acidification, because they have this innate ability to upregulate their pH through pumping protons out of their body into the surrounding. Now, we're going to take you back to boron. Remember, boron works because biogenic calcite takes up the borate ion, and the speciation has a pH dependency. So we can measure the boron isotopic composition and understand the pH. I won't get into the details of it. What I want to show you is that what is recorded by calcium carbonate versus what is calculated based on instrumental measurements, there is an offset. And the offset runs parallel to the expected line, which means there is a biological impact. However, the proxy holds, the thermodynamic validity holds, but there is a biological offset, and it differs from species to species. The question becomes why? So this particular calibration comes from this particular species called Orwellina universa, and you can see that it has a lot of spines. And among those spines, these are these bright structures, which are zooxanthellae, which are the symbionts, which are living with these organisms, which are doing the photosynthesis. We are supplying it with a lipid, allowing it to grow. So when the calcite grows, It grows at night when there is respiration by these symbionts. When the symbionts respire, they release CO2. When CO2 is released, the ambient pH drops slightly, and that is exactly what we are observing. So this observation shows the interplay between the symbiont and the host, which in this case is the foraminifera, and in terms of corals, there will be the coral aragonite structure. Now let's go back to the CO2 history. We have had this sort of CO2 fluctuations over the past, going between 180 ppm to 280 ppm. And boron isotopes in biogenic calcite, both corals and foraminifera, have faithfully reconstructed this. So we now know that we have a proxy which can tell you past seawater pH and atmospheric CO2 concentrations quite faithfully. Now we expect that with the rise in CO2 and the drop in surface seawater pH, corals everywhere across the globe should record this sort of drop in pH with lapse of time. Now, imagine this 1994 to 2024 window, we have a pretty rapid drop in pH. If you run an average line through this, which is going from around 8.15 to 8.05, which for calcite secreting organisms is quite a drastic drop. Now, this is a compilation of all coral records. I don't expect you to see it carefully, but I'm going to focus on this particular coral record from Lakshadweep. And this goes from the year around 1990 to 2013. Now, if we zoom into that record, what we observe that the boron isotopic composition looks like this. Remember, if CO2 rise caused a pH drop, the boron isotopic composition should have dropped. But we don't see an expected pattern in ocean acidification as you would expect from a coral which is living at the surface in a world where CO2 concentrations are going up. So we calculated what sort of pH drop should have been expected in that region. This is the curve for it. And this is what the boron is showing in terms of pH scale. So they're completely decoupled, which actually gives us a good indication that how resilient corals are towards pH change. And also regionally, oceanography controls how much CO2 gets absorbed into seawater and how much CO2 gets expelled out of seawater. So what we concluded that this particular region doesn't have the classical air-sea exchange system where CO2 is invading from atmosphere to the seawater. Now, but the coral bleaching is real. We are seeing a lot of coral bleaching. What's causing this coral bleaching? What's causing this coral bleaching is actually the rise in global temperature. Bleaching is not caused by ocean acidification or a decrease in what we call pH of seawater. It's caused by this increase in global temperature. As you can see, as we hit the 1950s and 16 onwards, the areas with red dominance starts populating this globe more and more. So there has been a significant rise in global temperature over the last few decades. 1917s onwards to now, there has been a significant rise. And what has that caused? The zoonotic, which inhabitate the surface of the coral, which actually is supplying the coral with the food. Remember, a bleached coral looks pearly white. So if a coral is looking pearly white, it has already, if they are photosynthesizing, you will have a different color. And the bleaching actually is these symbionts and invasive species coming in and getting rid of these natural symbionts which are supplying the corals with the food, because corals by themselves, these aragonite structures, cannot produce their own food. So it's a coupled system where there is a symbiont, a zooxanthellae, and a structure-forming organism. And if you get rid of the one that photosynthesizes, or you replace the one that photosynthesizes with something which is using this structure-forming organism as a parasite, then we have a collapse of the coral ecosystem. So it's a global warming which is allowing for invasion of these species or the decline in the photosynthetic rate of the natural symbionts, which is causing coral bleaching. So the take-home message from my talk is that corals are a faithful recorder. They are being damaged, and most of the damage is caused by how these symbionts are being, their productivity is going down, and how they are being invaded by other invasive species. So it's more of a biological problem from my perspective than that of an inorganic problem. Happy to take any questions.
Thank you, Dr. Mishra. That's really complex, and thank you for breaking down such a complicated problem for our popular audience here. Yes, we have 10 minutes for questions related to Dr. Mishra and Dr. Singh's talks. I don't see any questions yet.
I think Imrose has his hand up. Yeah.
Oh, sorry. I just
This is
yes, go ahead.
hi Shomoto. So yeah, so
Right.
this is so this is it's very interesting. So I'm just wondering, you showed that graph where there's a significant periodicity how the acidifcation is rising, right? What is determining that periodicity? It's very robustly periodic.
So this is quite interesting. This is the spring growth of the boreal forest in the northern hemisphere. So this is controlled. So the boreal forests are still, it's still debated whether they are net carbon sink or a carbon source, but they are still not a mature forest. So we expect them to be a carbon sink. And as you can see, the CO2 has this seesaw pattern. And if you zoom into that, you'll see that between May and August, the CO2 is dropping because the boreal forest is growing. And again, the leaves are shed, which gets respired and adding CO2 to the atmosphere system.
Okay, I have a question.
Yeah.
There's a lot of talk about resilience of coral reefs. So in really warm years, there's a lot of bleaching, but a lot of reefs also come back once the temperature goes to normal levels. And it seems that healthy coral ecosystems are really important for that. So is there a lot of study of resilience and the extent of resilience?
Not really, because you're right. As you pointed it out, it's the temperature which is causing more bleaching than the ocean acidification. That's because the symbiont is escaping. They have a very narrow temperature tolerance. So once you go above a certain temperature, they don't want to stay there. And above a certain temperature, the invasive species can take over. Mm-hmm. So if you have a resilient symbiont, which can go dormant and then come back up, then that coral will survive a lot of high temperature fluctuations. But if you have a delicate symbiont, then no, sorry, you won't.
Mm-hmm. Could you give an indication as to what these invasive species are? Are they like different invasive zooxanthellae or?
The invasive zooxanthellae, there's bacterial infection.
Wow.
So there is a lot going on, yeah. And that's something we have only scratched the surface at the moment. Yeah.
Meghna, go ahead.
Thanks, Ghazala. Thanks, Shambhu, for that talk. Very fascinating to see the details of how corals are working. I always look at it as more as an ecologist in terms of the manifestations of the coral in terms of an ecosystem. But I was curious, again, as coming from the ecology angle of things, I was curious when you talk of these, when we talk of bleaching and when we talk of resilience, I think one of the things that we need to think about, or we often think about as ecologists, is what is the diversity in the responses of these different. Are all corals, for example, or all zooxanthellae going to respond the same way? And you indicated that no, there are some zooxanthellae that have this ability to be dormant, whereas others will just eject themselves at the slightest hint of a warming time. So could you give us some idea perhaps of this diversity of responses that we see, and whether you have in your lab studied some of this as signals or consequences of adaptation to a changing climate?
I haven't. And I know the Western Australian group in Perth, they are doing this. I'm not an expert to talk about the biological side of it, I'll be honest with you. And what they are trying to do now is to go back to the bleached corals and populate them with zooxanthellae, which are temperature tolerant, and they have slightly better tolerance towards invasive species. So they're trying to regrow coral reefs, and they have had some mixed bag of success, which I'll be aware of. Unfortunately, in our case, the Andaman corals, we haven't even touched them. So we really don't know what's going on, and we really don't know what are the dominant species and where they're taking us. Yeah.
Sorry, just a very quick follow-up, and you haven't touched them because of, I don't know, logistical or administrative issues?
There's a logistical
or...
issue because you need ship time to go out there.
Ah.
International ships won't come here just to do some coral sampling, right? So and there is the permission required to do those things. So
Right.
there's a huge loop in which we are stuck. Yeah.
Absolutely. Yeah. Thank you.
Sure.
So also, I think, I don't know, this again from a very layman's perspective, but I see a lot of interactions from the microbes, because with this ocean water, as the temperature goes up, there are periodic infection by this Vibrio chiloe kind of bacteria, which kind of kills the zooxanthellae, right? So because what I also always wonder, because these corals, they have very strong innate immune response to this Vibrio, right? And when this pressure becomes very high, it's somehow, I don't know, I don't remember exactly what are those very critical chemical pathways, but there is some trade-off that often happens between innate immune response in corals versus their ability to do this calcium deposition. So I don't know if you have ever thought about looking at such trade-off at a ecological scale.
There is a huge, you're exactly right. Like, I have a finite amount of energy. If I'm spending it more on my immune system, I'm not spending it on my reproductive system and growth system. So the moment I start spending more energy on the immune system, my growth is stunted. And that has been observed very clearly in the lab, that the distribution of the energy is critical.
Okay, so yeah, Meghna has another question. Please go ahead.
Sorry, only if there's time.
Yeah, there is time. Yes.
Okay, my question was actually for Dr. Singh, if he's still there. I'm not.
So Dr. Singh has left.
Okay, never. Yeah.
But we can discuss from my limited knowledge of, if you want to ask a question, we can discuss it and then I can try to answer it. Or
Okay. Yeah, well, I was just curious whether there are any experimental studies that are being set up to monitor the impacts of tree line changes and these multidimensional factors, potential interacting factors that come into play with respect to the response to warming.
as far as I know, there are no experimental studies yet. Dr. Singh's recent study has been largely just, there's so little known about tree lines in the Himalaya that I think they were pretty much occupied for 3 years just documenting the species that exist and at what level. And then trying to, get some understanding from other systems. So yeah, he hasn't published anything yet on that. Yeah. But it would make for a really neat system.
Yeah, that's what I was thinking, that if it's possible, this would be a very good place to. I know that there are some warming experiments that people are doing in the, in the alpine regions on the sedge and herbaceous
area, right?
Yeah. But I wasn't sure, or I was just curious whether something like that was also being attempted here.
Mm-hmm. I think what's happening there is, I think the spillover of tree species into the alpine meadow areas, which is now one of the most common sites. And so that is going to really mess with the grassland ecosystems. But also, as you said, the different tree species show very different responses as well. So it's a complete disruption of what a community was at that point. But yeah, it would be really interesting because there are few species to study, so experiments would be easier to handle as well. So we can,
especially in terms of mycorrhizae, right? Because I was curious to hear more about that. Many of these species are probably ectomycorrhizae, which will be pretty critical in terms of their movement, or even in terms of their ability to survive indefinitely.
You think even at high altitudes there would be mycorrhizae?
I mean, many of these are temperate species, right? And ectos are mostly in temperate areas, and I don't think they're really temperature sensitive as such. I think like birches and
Fir trees. Fir trees have been studied a lot, yeah, in temperate.
Yeah, but also oaks, they are, many of them associate with ectomycorrhizae, and ectomycorrhizae can be very host specific. So that would be something worth looking at in the context of that elevational.
Yep. Well, thanks to both the speakers in this session. It was really fascinating. So let's go ahead. Sambuddha, would you have some parting comments for this
No,
session?
it was a pleasure. It was a pleasure being here.
Okay.
Hopefully we can proceed on this. Yeah.
Okay. So now I'd like to introduce the next speaker, who is Jessica Metcalf. Thank you very much for joining us from across the world. Very nice of you, and to bring us these insights from a very different angle on infection in a warming world. So let me introduce Jessica. She's a demographer with a broad interest in evolutionary ecology, infectious disease dynamics, and public policy. She completed her PhD at Imperial College on the evolutionary demography of monocarpic perennial plants. And then she has done postdoctoral research at the Max Planck Institute of Demographic Research and at Duke University. She has also worked on infectious disease dynamics at Pennsylvania State University and Princeton University. She's currently an associate professor of ecology, evolutionary biology, and public affairs at Princeton University. We're really grateful to you, particularly because you're coming from a really multidisciplinary perspective, and disease is, of course, one of the things on top of our minds right now, with warming and the zoonosis also becoming more and more prominent. So over to you, Jessica.
Well, thank you so much, and thanks so much for inviting me. Can you see my screen? Does that work? Great. And you can hear me all right?
Yes.
Wonderful, wonderful. Well, thank you again. It's been a great pleasure and some really fascinating talks around the topic of biology in a changing environment. As mentioned, I'm going to switch the focus onto infectious diseases. As everyone knows, am I sharing the screen with the notes on it or the screen without the notes? Can you see my notes?
Unfortunately, we can't see you.
No, you're good.
Excellent. For some reason, this is a new computer, so I'm getting used to it. So in early 2020, a coronavirus started spreading around the world, rapidly causing an international public health disaster of unprecedented scale. At the same time, in January 2020 was declared one of the hottest years on record, with land surface temperature anomalies as shown in the figure. So we have these two crises, one fast-moving and the other not as fast-moving, but still fast-moving, creating this extraordinary challenge to how we confront the years to come. Much of my work sits at the nexus of these two challenges, so thinking about how changes in climate and weather might change the burden of infectious diseases. And this can happen, of course, in many different ways, in part because so many different pathogens burden our populations. Turning our attention first to mosquitoes, increasing temperatures like those seen in 2020 and every year since then, increases the rate of life cycles of vectors like mosquitoes, enabling them to bite more individuals and thus spread diseases more rapidly. This is clear. It should be clear that this can result in much faster transmission of infection. So if mosquitoes are biting more people, then if the temperature increases by 1 degree, what you might observe is an increase in that transmission and therefore an increase in the burden of malaria by some proportionate level, depending on the relationship between temperature and transmission. However, of course, beyond a certain threshold, mosquitoes will no longer be able to survive if the temperature is really extremely high. And so a 1 degree increase in temperature might actually result in a reduction in the burden. Pulling together the evidence we have on the pattern of increases in temperature across the planet and what we know about the biology of the mosquitoes that transmit these sorts of diseases is one of the challenges in trying to anticipate what the burden of infectious diseases will be in a changing climate. And of course, temperature is not the only driver of such vectors. Rainfall is also terribly important. Rainfall affects both vectors, but also can drive events such as flooding. In 2019, diarrhea was estimated to have killed more than 1.5 million people on the planet. And this happens when viruses or bacteria replicate to millions within an individual, and then that infected person might excrete millions of these pathogens in turn, putting vast numbers of individuals at risk in the context of poor sanitation. A major battle in global health is to break these pathogen life cycles, and containing infected waste in effective sewage systems is a core part of this effort. Also, providing people locations to wash their hands, and all of these various threads enable us to stop pathogens getting into the next person. Clearly, any degree of flooding or breakdown in sewage systems puts these gains at risk. Vector-borne diseases and directly transmitted diseases that transmit through contamination of water are not so much the focus of much of my research, which tends to focus on directly transmitted infections. And here it's perhaps less obvious how climate changes might shape the spread of these infections from one individual to another. But it does happen. So here is some work from an MIT laboratory looking at how particles sneezed out by an individual spread through the air. And you can perhaps imagine that as the temperature changes or as the humidity changes, the ways in which these particles fall out of the air will be modulated. And this will result in greater or lesser transmission between individuals. One of the major challenges in this field in studying transmission of infectious diseases and the intersection with climate change is the fact that we never actually observe the event of transmission. We can see population level measurements of how incidence of disease changes across time. We might in some occasions have enough data that we can do household contact tracing to figure out the most likely timing of a transmission event. But in general, the actual precise moment where an individual acquired an infection is really not accessible to researchers. So a major thread in this research has been to investigate whether we could use population incidence data to quantify climate impacts on infectious diseases. This work really began with two brilliant graduate students at Princeton, Aisha Mahmood and Rachel Baker, and they were looking at some data on chickenpox in Mexico. And what you see on the left-hand figure, so chickenpox is a childhood infection that results in a bit of a rash. It's actually a reasonably mild infection, but it is one which is extraordinarily easy to diagnose, so there tends to be very good data on this infection. And what you see on the left is a pattern of mean humidity across the different districts of Mexico. And on the right-hand side is a measure that the middle map is a measure of the magnitude of transmission in different locations, estimated based on the average age of infection. If you have a sense of how old individuals are when they're infected, you have a sense of how fast that infection is traveling across a landscape. Because if it's moving very, very fast, individuals will get infected very young, so the average age of infection will tend to be low. And if you just quite simply put these two measures together, this measure of mean humidity and the measure of transmission, you see that there is a negative relationship. It seems that this virus transmits more effectively using this population scale data in environments, at least in districts where the humidity is lower. Of course, this tells us something, but it doesn't really put us in a position to predict the future, which is ideally what we'd like to do. We'd like to have a sense of how a changing environment might modulate the burden of infections like chickenpox. And so what we'd like to do is build a model of how transmission is happening. So to tackle this question, Aisha and Rachel turned to the fine-scale detailed data that was made available by the Mexican government. So they shared pretty much all their public health data online, which is extraordinarily generous and extremely useful for researchers doing this sort of work. And the figure here shows you for different districts in Mexico, shown as the rows with the names on the right-hand side, the incidence of the infection. So brighter yellow indicates higher incidence. So we now have a bedrock across many, many years, so starting in 1986 up to 2008, where we know how much chickenpox there was in every month of the year for all these different districts. And with this, we can put together a model of how the infection is moving between individuals. And if we have such a model, and we know a certain number of things, like once you've had chickenpox, you will never have it again. We know the approximate time that individuals tend to be infectious and how long it takes them to recover. So we can put these pieces together and try and fit this to the data. And of course, the question we're really curious about is the degree to which transmission is modulated by climate drivers. And one of the challenges here in general in trying to probe climate drivers in the context of infectious diseases is that everything goes up and down at the same time, right? So many things peak at winter, so many elements of human behavior tend to be extremely seasonal. So trying to pull out a climate signal in this context is rather statistically challenging. But if you have a mechanistic model, you can lay down a foundation and then you can start asking, what were in the years where, so in this example, there seems like there was a very high instance of the reported cases of chickenpox shown on the y-axis in the year 2000, whereas in the year 2001, there was much lower incidence of chickenpox. And so we can say, on the background of the seasonal cycle, is it possible to identify whether that year where there was very high transmission corresponded to much lower humidity than average, and the year where there was very low transmission was that much higher humidity than average? So we can actually probe the anomalies on the background of the seasonal cycle to get a mechanistic understanding of how transmission is modulated by the underlying climate drivers. And this is something that Aisha and Rachel did, and with this, they predicted future burden. So the percentage change is what's shown here in the colors for every month of the year on the y-axis across all the different districts. So you can see that there's like vast increases in some months relative to the background, and there are reductions in other months. So it's very interesting, and it was the required technical breakthrough in this area. It's also something that was not so interesting from a global burden of disease question, because as I had mentioned, chickenpox is a relatively minor disease. Rachel therefore turned her attention in this toolbox to a much more pernicious infection, and this is respiratory syncytial virus. It's the leading cause of under 5 mortality, even in places like the United States. It's, again, a respiratory infection that's transmitted by sneezing and coughing, and it has this tremendous burden of morbidity and mortality. We also have a rich array of data. So this is time series from different districts in America, different provinces of America, showing you that with varying, so that the x-axis is of various lengths in each time, but what you're seeing every point is a measure of the number of cases reported for that particular county. And you can do the same basic thing. You can start sort of probing the course scale patterns of these time series to see whether there is a reason to suspect that there might be climate drivers underlying this. And so this is how Rachel started. She plotted temperature across all the different counties in the United States, specific humidity and precipitation, that's the 3 x-axes, against the onset week. So the first week in which you saw an increasing number of cases within the year. So counting weeks from week 1 to week 52 in the year. And you see this very strong relationship. So where the temperature is really low, it looks like respiratory syncytial virus tends to start later in the year. And when the temperature is higher, it tends to start earlier in the year, and the same basic pattern for specific humidity. So Rachel used the same tools that she'd used for chickenpox to put together a mechanistic model to actually probe exactly how much transmission might be modulated in every, I think she had week of the year in this particular instance, to see whether the anomalies could inform the degree to which you would get a 1 degree increase in transmission, perhaps for a 1 degree increase in temperature or specific humidity, or rather a decrease in this instance. And with this, again, you can start predicting out the future. So you could predict the pattern of infection expected starting in, so this is comparing what you get in the next 50 years versus in the year 2100 onwards. And what you see that as time moves forward, you end up with a much flatter relationship. So the blue lines indicate the further into the future projections, and they tend to be much flatter, but with shallower troughs as well. So this might shift how you made an investment in your healthcare facilities across the course of the year, if there is going to be the shift in when the burden manifests itself. So this is interesting in itself and lays the foundations for understanding what the future burden of disease will look like. It also yields sort of interesting underlying mechanistic understanding. So both for influenza, which had been done by a number of other research groups in previous work, but also for RSV, respiratory syncytial virus, we see this U-shaped relationship with specific humidity. So transmission is very high, either at very low or at very high specific humidity, based on the statistical models we developed. And this lines up quite nicely with work that occurred during the SARS-CoV-2 pandemic that pulled together a mechanistic understanding of how viral sort of elements in the air might survive, what their half-life would be across a spectrum of relative humidity. So with these pieces in play, we can start to lay down predictions for what the future burden of infectious diseases will be, things like RSV, things like chickenpox, and this might lead us to reconsider how we design investments in healthcare across the year. I've been skipping over one piece of this, which is that there are two elements of clockwork at play in this particular instance, and two elements that need to be taken into account to do this effort of estimation. So to illustrate this, I'm showing you a very iconic time series. This is the time series of measles in London, starting in 1945 and going all the way through to 1965. And the kind of driving clockwork we've been talking about is the fact that in many parts of the world, you get these very regular seasonal cycles where things are cold, for example, around Christmas, or warm if you're in Australia. And then during the summer, things are warm or cold accordingly. And so what we see for something like measles might be that you get these very strong peaks in winter that are sort of driven by, in this particular instance, it seems to be more human behavior than the climate drivers, but there is a strong seasonal driver that results in these particular peaks at particular times of year. But if you gaze at these time series, you might be seeing that there's something else going on. And that something else is that as you move from the left-hand side of the time series to the right-hand side, we shift from annual dynamics, so an outbreak every year, to biennial dynamics, an outbreak every 2 years. And what's happening here is that right after 1945, which was right after the Second World War, there was a baby boom. So the number of births coming into the population increased, and this provided more susceptibles that could spread the infection more rapidly. So you also have this dynamic that's this susceptible clockwork that's also driving the infection. So as numbers of susceptibles are coming in, you can have a big outbreak, there's then depleted by infection, another one as new births come in, etc. And as the birth rate falls after the baby boom, there's not quite enough births to allow a second peak every other year. And so you end up with these biennial dynamics. And it is crucially important to take this into account when trying to project the burden of climate and disease. And to give you an illustration, when we were thinking about SARS-CoV-2 in the summer of 2020, many people were saying, well, you know, summertime, it's a winter virus like so many of the other coronaviruses, it will just go away. And remember, we knew so little about the virus at this time, at this point in time of the pandemic. What people were expecting was the dynamics I've shown here on the right-hand side of this figure, where you get these regular oscillations. The lower solid line is incidence, so peaking around Christmas every year, around the peak of winter every year. And then above that, I've shown susceptible individuals oscillating as well. But what is missing is the fact that we were not in the endemic phase where you would get these regular outbreaks. We were in the emergent phase where you get this first very large outbreak, very large number of cases to begin with, as everybody in the population was susceptible, which drives down incidence, and so you get a much slower increase in susceptibles and a sort of non-regular oscillations for a little while. You can show this on a phase diagram plot. So on the x-axis here is the proportion of the population that's susceptible, and on the y-axis is the proportion of the population that is infected. And you start off with everybody being susceptible, and then infecteds increase through time, and susceptibles are depleted because everyone who is susceptible and becomes infected is no longer susceptible, so can no longer be infected. And then you slowly move in towards the attractor of these dynamics. But the main point is that what you will see over those first few years is certainly not a simple disappearance of the pandemic during the summer. It will really be governed by the susceptible side of the equation as well as the seasonal driver side of the equation. So the whole dynamics are likely to be much more complicated. Another interesting part of this, of course, there's many other pieces in play. This was really just a thought experiment to demonstrate that assuming that the virus would disappear in summer was rather naive, given what we knew about the underlying susceptible dynamics. In reality, there was also two things to contend with. one of which is the emergence of new variants, which meant that the ways in which susceptibles were manifest was much more complicated. And the second was the occurrence of non-pharmaceutical interventions. But non-pharmaceutical interventions themselves also illustrated the vast importance of susceptibles and taking account for susceptibles, not just for SARS-CoV-2. And when I say non-pharmaceutical interventions, I should say this refers to all the efforts, all the things that were referred to as lockdowns or closures of schools or preventions of gatherings, which resulted in less contact between individuals and thus less transmission of infection. So if you looked, for example, at cases of influenza across all the different states of America, so the states are shown in rows, and the color shows the proportion of cases in every week of the year relative to background. The moment the national emergency was declared, around the 11th week of the year, the number of cases of influenza also collapsed. And this is because as SARS-CoV-2 could not transmit between individuals if individuals were not in contact with each other, this also was the case for all the other directly transmitted infections like influenza. So interesting, but also something that instantaneously raised alarm bells amongst people who think about the infectious disease dynamics and susceptible depletion. And that's because if no cases are occurring, as shown on the cartoon on the right, the number of susceptibles is going to build up. So the number of individuals who haven't had the infection will increase and increase. And so what you might expect to happen is that you would get a very large outbreak in the wake of this. And this indeed has occurred for both for respiratory syncytial virus and influenza and other of these directly transmitted infections. One of the ones we've been working on a lot, and this is the work of Daniel Park, a graduate student at Princeton, is enterovirus D68. And we predicted that there would be a very large outbreak following the release of the non-pharmaceutical interventions around 2022. And this is indeed what happened. So that's to talk about the ways in which climate drivers might shape infectious diseases. I think it's really important never to neglect the fact that the human element is also incredibly important. So the ways in which the burden of infectious diseases will manifest across our populations will, of course, be a function of the ways in which climate varies across the globe, but also where people are. So this is a map projecting population density in 2100. And if we want to understand the concentration of and the spread of infectious diseases, we need to consider these two sides of the coin, both how the climate is shaping infectious diseases, but where the people are that the infectious diseases are spreading between. So human density itself will be important, but as will human mobility. And human mobility is one core way in which people move infections around the planet, usually without intent, obviously. And this also shows very strong intersections with climate. So we know that climate disasters can drive human mobility. We've done some work in Madagascar, for example, using mobile phone call data records to model the movement of people across populations. And you can show that districts or communes where cyclones have moved through show a net loss of individuals as people leave away from this, and this can end up aggregating diseases in different locations on the planet. There are, of course, also places where people move seasonally, driven by climatic variation in resources or, agricultural processes, or there may be sort of just regular movements of this kind. But what I'm going to focus on next is not so much human density and human mobility, but how climate might actually disrupt control efforts themselves. And again, I will be focusing on the question of cyclones, but in particular, the intersection between cyclones and malaria incidence. So this is a map showing the incidence of malaria per 1,000 population, so in the red colors, but also showing the trajectories of cyclones across the planet. And you can see that there are many, many, many cyclones in particular strike the island of Madagascar, which is also a location where there is a very high burden of malaria. Benny Rice, a postdoctoral researcher at Princeton, was working in Madagascar on malaria at a time when two different really major cyclones hit the island, Cyclone Freddy in 2023 and Cyclone Batsirai in 2022. The results were absolutely devastating. Populations suffered tremendous disruptions, and obviously all efforts that were in place to deliver healthcare to individuals and to deliver, in particular, clearance of malaria. So malaria is a tremendous burden in this part of the world, and malaria is something which has a very high mortality rate and morbidity rate. And efforts, as you can imagine, in the wake of a cyclone to deliver prophylactic drugs or to clear the infections are going to be extraordinarily intractable. Benny worked across 2 years in 10 different sites in these areas before and after these particular cyclones. So this is just a visualization of the data he obtained. Every one of the different blocks is a different site, so of the 10 sites, sort of rectangles. And every row represents a different individual measured at the different time points shown on the x-axis. So there's sort of something like 20,000 data points in there. And at every time point, what he did was he came back, he tested all the individuals within the populations. If anyone had malaria, he tested individuals using a rapid diagnostic test. If anyone had malaria, they were given the drugs to clear the infection. And then, he came back again 30 days later, and you could see the degree to which individuals were acquiring malaria over this time frame. So this is already really unique data and a unique lens onto the rate at which individuals become infected. So how fast the interventions that you've put in place are eroding. But it also, since we have data around the cyclones, gives us a sense of how disruptions driven by cyclones could negatively affect this effort to intervene. And so if you imagine that we come in and we treated everybody in the population where the vertical line lies, and you give everybody a particular drug that clears their infection and also protects them from some amount of time, in different locations within his study site, people reacquire the infection at different rates. And the rate of acquisition of the infection can be described using what's called the force of infection. So you can think of it as the underlying risk that people are infected. And across our study site, we had 3 different scales of the force of infection. So there were some places where individuals will get reinfected almost immediately, that's shown by the yellow line. And there are some places where individuals will get reinfected much more slowly. But in all of these locations, pretty much, you really had to come back to the populations really quickly if you were targeting, for example, maintaining the rate of infection of the population below 10% or something like that. One of the main sort of insights from this work is we know that our efforts erode. We know that the efficacy of the drugs against malaria will decline over time. But one of the most exciting pieces of news in global health recently has been the development of a malaria vaccine. So longer half-life drugs would give you longer protection, so you get a slower increase in the probability that individuals were infected. But if you had vaccines that could last even longer, this gives you so much more time to return and intervene and keep the population below that threshold of infection. So what Benny did was, using all of this data, he both explored the potential for different drugs to maintain levels of health within the population, but he also explored the prospects of introducing vaccines into the population. So this landscape shows you the percent fully vaccinated at the time of a disruptive event. So this might be a cyclone, but it could be also other forms of disruption. And the y-axis is the percentage reduction in symptomatic infections. And he made various assumptions about vaccine efficacy, etc. But really what you can see that the more people you can vaccinate at the time of a disruptive event, and the greater the efficacy of the vaccine, the greater the reduction in symptomatic infections. So these have tremendous power to carry us through. We know that the frequency of things like cyclones is likely to increase, or at least the potential disruptiveness. And there are ways in which we can design interventions that can carry us through this. So I talked a little bit about directly transmitted infections and their future burden, a bit about the ways in which disruptions of control could shape our future. But there's also, I think, a really interesting set of questions around how infections of other species on the planet will affect our future. In particular, thinking about food security, plant pathogens are perhaps a neglected area where climate disease could play a really important role. And to investigate this, because we wanted a large variation in climate in order to grapple with this, we couldn't unfortunately go to the Himalayas, or that would have been wonderful. We worked in the Rocky Mountains, looking at a vast temperature gradient across a vast altitudinal gradient, focusing on 4 different sites and looking in particular at a plant pathogen that was extremely easy to identify. So it is this flax rust. So the plant we're focusing on is this flax plant, and the pathogen is this little fungus called the flax rust that you can see as the orange pustules on this figure. And this entire research program was designed and implemented by a graduate student at Princeton, Ian Miller, who also monitored all of the climate variables you might be interested in. The legacy of Ian's first work, showing how these different drivers might shape the spread of infection, was followed on by Keenan Dougal, currently a research assistant in the lab, who has put together the trajectory of the outbreaks across the 5 different years for which we now have data, shown in the different colors. And he noticed that something really strange was happening in that lightest blue line in 2023. Whereas you got these really nice saturating curves as days passed since June 1st, you see an increase in the proportion infected, but in 2023, you see this sudden collapse. And if you compare the dynamics relative to other years, you see there's like a real loss in the percentage of infected individuals within the populations. So Keenan put this together with the data that was available on climate variables, and showed that really the really strange thing about 2023 was that it was a spectacular drought. And what seems to be happening is that during the drought, the plants actually drop their leaves to maintain their survival. And by dropping their leaves, they get rid of the pathogen. So the pathogen is in some sense much more sensitive to the drought than the plant is itself. There's been a lot of modeling and thinking very broadly about how climate change might shape the spread of plant infections. And much of this is focused on temperature and has suggested that plant pathogens might spread further and faster with temperature. But this gives the reverse side of the coin, which suggests that for many plant leaf pathogens, you might actually get a really serious impediment to their spread associated with phenomena like drought. And by studying these sorts of high altitude environments, we might get a window onto how the future of plant pathogen trajectories will play out across the future of different climates. So I've given you a whistle-stop tour of some of the work we've been doing on the transmission of airborne pathogens, in how disruption of control efforts by climate disasters might shape the future landscape of disease, and then a little bit on our work on plant pathogens. I think so much of this, we still have so many knowledge gaps, and in order to address these knowledge gaps, we need huge amounts of data on the incidence of infections in people and in plants and in animals, alongside really good climate data to tease apart what's actually going on and use dynamical models to predict what this will do in the future. We're very lucky at Princeton in that we sit very closely to a group of excellent climate scientists. So Gabe Vecchi, Wenchang Yang, and Maya Chung, shown here, are part of our climate and disease group at Princeton University, who work on and around all of these different topics. So thanks very much, and I hope I didn't go too far over time.
No, thanks. That was really interesting. It was definitely a lot to take in on very different diseases and their transmission and so on. It's, as usual, pretty complex. So we'll take some questions for Jessica's talk right now and then go ahead to the last talk. Yes, Meghna, go ahead.
Hi, Jessica. Am I audible? Thank you so much for the talk. It was a really fascinating tour of the kind of work you've done. And as I'm a community ecologist, and I study, or a part of my work involves understanding how plant pathogens play a role in maintaining plant diversity. So I was curious from that angle. You spoke about this one disease and tracking the incidence or the epidemiology of this one disease or one pathogen across an altitudinal gradient. But clearly, this is going to be one of many diseases that different plants harbor or would be susceptible. Have you thought of or looked into the interactions among different pathogens, or how pathogen communities themselves would change directly or because there's a change in plant communities with climate change across an altitude?
This is a wonderful question, and it's something I realized I forgot to say. That there is the interest in plant pathogens from a very pragmatic maintaining food security side. But of course, we know that they're probably incredibly important at maintaining biodiversity and maintaining diverse communities. There's a famous theory of the diversity of tropical forests, the Jansen-Connell hypothesis, which suggests that you can get this coexistence diversity of trees because you have different pathogens, each of which specialize on the different trees. And so you end up with a situation where if you are close to your parent, you're disadvantaged because your parent carries that pathogen. So you end up with this, this sort of apparent competition effect. I had a wonderful former postdoc at Princeton wrote this paper suggesting that, in a way, by laying out the Jensen-Cole hypothesis, you're just kind of shifting the problem because it's not clear what maintains the diversity of the pathogens, right? So I think there's a lot of really interesting remaining mysteries there. And of course, we need theory, but we also need data, and perhaps we need data more than anything else. And so I'm really fascinated to look at your system as well. I don't know very much about systems where we have a really good handle on the diversity of microbial communities on plants. And it's particularly complicated, of course, because they're not always bad. Many of them are also very good. So
absolutely,
I actually suspect that you know much more about this than I do.
Yeah, I was just curious. No, I think the documentation of plant, microbes on plants is just happening over the last, let's say, half a decade or so, with the advent of next-gen sequencing, things like that. But this might be something that, across altitudinal gradients, for example, it might be something worth considering, especially this balance of generalist versus specialist pathogens, and how they shift with a climate shift in the climate.
Absolutely, I think it's an exciting time, and the data is starting to come in. So yeah.
Yeah, Imroz, go ahead with your question.
Hi, Jessica. As all, I think that so. Yes. So this is so I think you mentioned one part where you said that the shading of the leaves, which kind of puts pathogens under more exposed to climate change. Means, are there any specific parameter that you should be looking at from the animal pathogen as well? What distinguishes this under this particular context? The climate change will be more harsh on plant pathogens versus animal pathogens.
It's a really interesting question. So and it probably depends on the type of animal as well, right? Homeotherms versus ectotherms. I'm not with the degree to which some of us maintain our temperatures better than others. I'm not aware of. I'm sure people have thought about this in the larger comparative context, but I don't have that at my fingertips. But I think it's a fascinating question. I suspect again, Imroz, you might have better ideas than I do on this topic. You're muted.
You're muted, Imroz.
Sorry about this. Yeah. So no, I was wondering whether people are actively looking into the aspects where how this pathogen is evolving. So such contexts, right?
When we started this work on this plant pathogen, we were really interested in seeing how climate drivers might shape the evolution of virulence. And we were disappointed to learn that our pathogen was not very virulent as far as we could identify. But it is a very interesting. I mean, I think there will be a lot to be done in that space as well. I think that it will be, as Meghna mentioned, that I think a lot of our learning will come from wild plant populations, because what happens in agriculture is you see the pathogen and you get rid of everything. So some of these nuances of how different selection pressures, how the different scales of growth and trade-offs might play out might be missed. But I think that, yeah, absolutely. I'm really curious about that and really interested to see how that plays out.
And I think my second question was, sorry, the last question is will be on your segment where you've been talking about vaccines, right? I think this may be a very common question you might have faced maybe a million times. So unlike vaccines which are kind of sterilizing, like polio, right, which kind of eradicates the pathogen completely, most of these vaccines also tries to reduce the disease symptom in a way which might make pathogens to be more tolerant towards the vaccination. Do you include such parameters in your model when you look into the vaccination part of it?
So this is a reference to a really beautiful thread of research into thinking about how vaccines that separate transmission from disease might result in selection for greater disease, because the vaccines kind of can allow themselves to be more transmissible in vaccinated hosts because the hosts aren't dying as quickly, effectively, so they have more time to transmit. So you can get evolve greater virulence. I think that there was a recent lovely paper involving Western India that made the point that in identifying conditions where this will happen, we need to know a lot more about the underlying mechanisms, of course, which your work has contributed a lot towards, and the exact underpinnings of that trajectory of infection. We did some work thinking about the pertussis vaccine. The acellular pertussis vaccine there's a suggestion that it operates in this way, but it's really unclear and things probably move relatively slowly. I think that it's absolutely the case that evolution of virulence can happen in response to vaccination. We know this from Marek's disease in chickens. I know of no really good example in human populations. I know of many, many threads of data I would love for us to have in order to make these predictions more robustly, but it just is really hard to work out the trajectory within host infection versus burden, and unless you're working on a beautiful beetle system to actually try and unpick what we might expect the patterns to be. I think there's a lot of lovely work to be done on mapping out the possible landscapes, and some of this, for example, Rustam Andia and JJ Bull have started towards. But there's no lack of work to be done.
Yep.
Okay, thanks a lot, Jessica. We'll take more questions after the last talk. We'll have 20 minutes to take questions for the whole panel. So Meghna, go ahead and you can start your talk. I wanted to introduce you.
Okay. Maybe I can try sharing my screen in the meantime.
Yes, please. Yeah. So Meghna's, it's great to have her here. She's an old friend and colleague, and she's done amazing work during a PhD on rainforest ecology and regeneration from Yale University, United States. She's currently working as a senior scientist at the Center for Cell and Molecular Biology in Hyderabad. She works on the ecological mechanisms underlying community diversity, as well as the anthropogenic effects on the mechanisms that maintain diversity and restructure ecological communities. She combines ecological theory, experiments, and observational field research with advanced statistical models, which is, I think, a rare combination. She uses forest plant communities as model systems. Today, Meghna is going to speak on plant communities in relation to climate change. Go ahead, Meghna.
Give me 1 second while I just want to check if you can see my screen. I was having some.
Yes, we can, and we can hear you. Yes.
Okay, great. Thank you so much. And thank you for that introduction, Ghazala. Well, today I will not be talking a lot about my own work, actually. I thought that, this is an opportunity for me to actually look into some of the other kinds of work that are going on, encompassing a broader suite of ecological questions that people around the globe have been asking. And also, when I learned about the kind of audience that we are likely to have here, which is a popular audience, and you said that this talk would be hosted perhaps on YouTube, I thought I will take this opportunity to make this talk more general and just simpler. So I won't be having a lot of graphs, definitely not from my own work. Perhaps I will share that at a later date, and I'd be more than happy to. But today we are just going to talk about how biodiversity seems to be responding to the massive changes that are happening on planet Earth. And what are some of the signals that we have been seeing, and how we as scientists are looking for ways to understand the best ways to project what the future of biodiversity might be with the changes that we anticipate. So while I was preparing for this talk, I came across this very nice graphic by NASA. This deals with the average temperatures, global average temperatures in a monthly manner, starting from about 1880 onwards. And as you can see, everything was fine. We were hovering within the normal or the averages that we have seen for the longest time until about the 1960s, 50s, 60s. And then things started to spin out of control pretty soon. And today, as I sit here in the city of Hyderabad experiencing one of the hottest summers that I have ever had in my life, this seems to be an appropriate graphic that tells us what is happening across the globe, what we are all experiencing, where the temperatures are getting much warmer than we have ever seen them. And while this is talking about a global average of 1 degree, local changes in temperatures, like what we saw in the talk that Dr. Singh gave about the Himalayas, can be much higher, and the impacts of it can be felt quite strongly. The reason why we are having this massive shift, this massive change in temperatures across the planet today is in large part because of a human influence, the amount of carbon dioxide that we are pumping into the atmosphere, but also how this changing climate is resulting from a plethora of changes that humans have imposed on the planet. This involves the destruction and loss of habitats at massive scales, degradation of remaining habitats, or fragmentation, which breaks up large chunks into smaller parcels that do not quite maintain the kind of the processes that we've seen in ecosystems before, and a whole lot of other issues that accompany many of the changes that we today see or together see as global environmental change. Now, in this era of massive and dramatic changes to the planet, life is becoming hard for many of the species and organisms that share the planet along with us. Because of rising temperatures, many ecosystems and species are experiencing drought, untimely fires. There are massive die-offs of species, shifts in their ability to stay in some places or move through places, such as human-dominated landscapes where the habitats have gone and the remaining habitats can have very different climatic characteristics, and the details of some of which I'll get into soon. And some species, we know now from evidence, are just not able to make it. And we know that there have been these records of massive die-outs of individuals in some species, like 45,000 flying foxes dying in 2014 because of a heat wave in Australia. And the more sad cases of, which I feel sad about this, of species which are going extinct in the very recent times, whose extinction is attributable directly to climate change. A poster child of such an extinction is the golden toad, which blinked out of existence in 1989. And this was because its home in the cloud forests of Costa Rica simply got too warm to maintain the clouds that are typical of these areas. And the frog couldn't make it through this change of habitat. The bramble K. melomis is another very unique rodent species that lived on a single small island between Australia and Papua New Guinea. And it went extinct in 2016 because of sea level rise and change in cyclones and storms that flooded its home, killed food plant, and destroyed its nesting sites, post which its population just could not recover. Now, of course, these single instances of species going extinct is sad, but when we change or move from this story to the statistics, the picture still remains quite bleak. Analyses that have pulled together data from many, many different sources seem to suggest that regardless of whether we are seeing a 1 degree change projected in the future or a 4 degree change, most areas of the globe are likely to lose anything between 25 to 75% of species that we see today or that have historically been there in these areas. And this is a tragedy in the making. And as scientists, we try to understand, is there any way we can predict how different species might be affected by these massive changes that we see around us? What are the properties of features of species or of systems that we can use to forecast some of these changes that we are anticipating? And towards this goal, we often rely on something called traits, which are the inherent features of organisms that relate to their form and function. And functions are often important in terms of where the species is going to be found, what is the range of space that it can occupy, how well it does in different kinds of conditions, because these inherent features that are relevant to its physiology and function drive how it's going to respond to macro and microclimatic variations, such as water availability, temperature. And if we have this baseline information, this database that will allow us to look across species, the suite of adaptations that have evolved for different kinds of environments that we find at small or large spatial scales, we might be able to predict the fate of different species in a future condition that we anticipate. And in this graphic, it illustrates this attempt in trees, which is a taxa, a group that I study, and people in my lab study a lot. And just as an example, for trees, you have these features of their leaves, of their wood, of their roots, how thick the leaves are, how rapidly the tree photosynthesizes, how thick or dense the wood is, what is the kind of machinery that the tree has for moving water through the wood, how deep its roots are. All of these traits, either individually or in combination to make up the phenotype of a tree, can be relevant towards predicting how the species or this individual might behave in a certain given context of, say, temperature or water, right? And we use some of these traits to first understand where these kind of species are found in nature. So scientists have seen, for example, that as you go, if you look at the graphic, as you go from low to high water availability, you see a shift in what kind of traits become more and more common in a community, and they are commonsensically, almost intuitively related to what are the most important physiological functions or adaptations that species in that kind of context, climate context, needs. So if you look at places that have very low water availability, and you relate that to a trait which is to do with the hydraulics or how the species moves water through its system and how much water a species might use in order to maintain its photosynthesis and its function while it's doing its thing. We see that most species tend to have traits that are what are called slow and safe. They're not using a lot of water. They're slowly using whatever it is, being very conservative. And very, very few species are able to have a risky lifestyle. Maybe they're restricted to some small pockets of microclimate where water availability is sufficient for them to use a lot of water. And you see that this kind of the profile of traits in the community changes quite drastically and quite markedly as you go from low to high water availability. And these understanding of species features have been useful in also understanding what kind of species are at risk in a changing climate. We have so much data now from long-term monitoring plots across the world where we are seeing mortality of trees in response to extreme drought years, extremely hot years, and we are seeing clear correlations that when you are shallow-rooted, when you're unable to access water, or when you need a lot of water to survive, that is when those are the kind of species that are going to be hit the hardest in a drought context. And this information is helping us then project what the future of these ecosystems might look like, whether the composition of species is going to change in these ecosystems because some species are going to be winners and losers, which alter the future trajectory of the system. And not just forests, but this kind of understanding can also be extrapolated or is used in many other systems. We saw this really interesting talk about corals and coral reefs, and we've heard about coral bleaching. But even when corals are not subject to a dramatic change such as bleaching, rising temperatures can alter the diversity of life or the community of fishes, for example, that we see on corals. And this can be explained by the thermal affinity of the fishes, which is a product of evolution that has happened over millions of years as these organisms have been through or have been selected across millions of years of evolution. And what these studies are showing, in this study, for example, they looked at how thermal affinity might explain the change in the abundance of fishes after an extremely dry year relative to the baseline abundance before that year. So on the y-axis, any value that is positive means that this fish species has increased in abundance, and anything that's negative means that after the drought year or the extremely warm year, the species has gone down in its abundance. And the data seems to suggest that there is a signal that species which are more affiliated to warmer conditions tend to be okay or do better after this extremely warm year, whereas there is a decline in species that prefer to affiliate themselves with slightly cooler conditions. And these kind of trends are being seen from many places across the globe in marine ecosystems, but also from terrestrial ecosystems. And even properties within species can drive, like where individuals within species are located, especially when species are wide-ranging, can drive what kind of shifts we might expect to see in those species in terms of where they will be found in a future climate condition. So in this paper, where they looked at the phenology of nesting or the behavior of nesting in species, because nesting is very closely driven to temperatures, especially in temperate birds. And the study was done in the for North American birds, breeding birds. And each panel here shows whether the temperature for an individual that is sampled for that bird is cooler or warmer than the mean temperatures where usually this bird is found, right? And on the x-axis, a positive value suggests that a year that has been warmer than usual in that particular temperature zone of this range, or a year that has been cooler than usual. And what has been the nesting success in these conditions at this particular climate context? And what they found is that by and large, when the bird is in an area that is cooler than its mean temperature where it's found across its range, they tend to have better nesting success on warmer days. And this trend flips when the bird is found in parts of its range that is warmer than the average of where it is. So which suggests that if things become warmer and warmer, these species or these individuals are probably going to get knocked out of these sites, which are already the warmer ends of their range, because they're just not able to reproduce successfully and maintain their populations. Whereas there might be some reorganization of the community depending on how well some of these birds do relative to other birds, the cooler end of it. Right? And these features of species, these inherent traits together can help us predict what kind of changes we might see in the community. For example, we saw in the other talks how communities might change across an elevation gradient. And these traits can help us, for example, the thermal affinity traits can help us predict which species across, when we're going from a low to high elevation, how the community at a higher elevation might change, because some species that are very sensitive to temperatures might drop out, and others which are found in the lower elevations might actually be able to expand their range and occupy a space that was previously unfavorable to them because the climate was excluding them from it. And so we need more and more of such studies which are sort of bringing together large biogeographic patterns with smaller scale changes to communities to understand how different systems and different taxa are going to respond to the changes in climate that we are seeing around, and to project what these future assemblages look like. And most of what I spoke about were like within a community, but changing climate can actually lead to what we think of as a trophic collapse, or a change in the ecosystem from a food web perspective. And this study is a great example of that, which looked at the impacts of warming-driven decline in insect populations on what happens to organisms or animals that are dependent on insects for their food. And what they found in this forest in Puerto Rico, in Luquillo, was, years, warming-driven decline in insect population was closely tracked by a decline in the especially those that were dependent and for insects that was happening in this system because warming was affecting one trophic level, which was the food for the next trophic levels. And so you're going to see a lot of biodiversity loss, or you're likely to see, in fact, they have seen a substantial biodiversity loss in this forest because of the loss of one layer or one group of organisms that have been affected first by the climate, even though the climate did not directly affect the other animals whose abundances have gone down. But changing climate, especially the drastic changes we see today, can scale up to have effects at the level of the ecosystems themselves. For example, we have been having these massive droughts that are happening and large-scale die-offs and mortality in trees. The Amazon forest is a great case study where of a system that generates its own rain, that generates its own climate. When you have a massive drought that hits this place and trees die, as you overlay another stressor like land use change. Here we are seeing that forest that has been lost because of slash and burn agriculture or generally destroyed because of to make way for human needs. And when the forest area reduces, there is less and less forest or fewer trees there in the system to generate the kind of water that is needed for it to maintain the wetness that the Amazon is known for. A drought in that year then is experienced more severely and leads to more severe consequences, increasing forest fires, increasing tree diebacks, and shifting this forest to an alternative state, which is of a more open savanna state. And this is happening in many places around the world, which is called the savannization of forests, because of this interplay between more multiple stressors that interact with the climate to shift systems into a different state from what they have been for a very long time. And land use is a driver which is known to interact or play out interactively with the climate in terms of shifting what kind of communities we are likely to find in an area. And also it's going to drive what or how species will respond in these human modified landscapes where land use has changed the system from what it used to be. And this is actually an angle of research that we are very interested in, something that I have worked on quite considerably, and something where, a lot of the research in my lab is going, is to understand whether land use, which can degrade or fragment forests, results in a different impact of a changing climate in some parts of the remnant habitat versus other parts. And the rationale for this is that if you take a forest, for example, a closed canopy forest, and you have an edge which is often warmer and drier than cool, humid interiors. And this is fine as long as you have a big patch of forest where most of it is closed canopy, cool, humid interior conditions. But when you start chopping it up, because of land use change, you break it up, you increase the amount of edge that is there. And now you have sites that are much warmer on average than, and would be favorable, therefore, to species that are, or would be more conducive to species that are adapted towards warmer conditions. Now overlay this with a dry year or a drought that happens, and species or organisms that are there at the forest edges are more likely to experience a more severe drought, imposing a double whammy that filters species, as opposed to what the impact of drought might have been if you had an intact forest. In fact, data from across the globe is showing that regardless of whether you are in a wet, humid tropical forest, or if you're in the boreal forest, having an intact canopy cover can buffer the kind of temperature ranges that you find. So in warmer conditions, the temperature under the canopy can be nearly 5 degrees lower than what you find adjacent to it. Whereas when you go to a warmer site, the temperatures can be 2 to 3 degrees warmer, making it somewhat more comfortable for the plants that are there, as opposed to not having canopy cover or having a fragment, the edge of a fragment where microclimate tends to be a little harsher than what you see in forest interiors. And this is a line of work that we are taking forward in our lab quite deeply, and focusing on the younger life stages of plants. A lot of the research on plants and their response to climate change has predominantly looked at the older life stages of trees, because they store a lot of carbon, they're important for function, and if big trees die, you lose a lot of carbon, leading to these kind of feedbacks, negative feedbacks that can be quite detrimental to ecosystems and increase the sort of impacts of global warming or climate change that we're seeing. But we like to look at the seedlings, because that is what is telling us what the future community is going to be. Which species are likely to be winners and losers in a future climate. It's also nice that seedlings are tiny and therefore more amenable to experiments, which is an approach that we like to take in our lab, combining a whole lot of observational data, which will hopefully be long-term, with experimental data that lets us tease apart some mechanisms. So in this case, for example, we have tagged over 8,000 seedlings across this landscape, and we have been monitoring them for the last 3 years to understand across a gradient of microclimate, which is of soil moisture availability or light availability, are there certain features of these seedlings, particularly concerned to their, related to their ability to use water or tolerate drought, which might explain where these seedlings are found and which parts of the landscape are they best suited to. But also, imagine that we track these seedlings through time, and you have a dry year and a wet year and another dry year, or the extent of drought changes from year to year. This information is going to let us track which species end up being winners or losers with those changes or shifts in climate from year to year. And with that, we can eventually model and try to predict what the future community might look like, given that we have a certain trajectory of climate. This is work in progress, and maybe at a future time I can share some results with you. But really, this kind of work requires you to maintain these observations going for 10, 12 years to get some kind of time series data that can show you what the projected future trajectory might look like. But I spoke a lot about climate, and I spoke about how individual organisms respond to the climate, but ecosystems and ecological communities are also known for their interactions. Organisms interact. That's what makes ecosystems complex and interesting. Plants also, they might stand in one place, but they have a myriad of interactions with birds that eat their fruits, with insects that chomp on their leaves, with fungi and other microbes that are there in the soil. And microbes in particular, we are increasingly understanding, play a critical role in driving the dynamics of plants. For example, the competition between 2 tree species can actually be driven by what kind of microbes they harness. And I won't go into the details of this, but this is a key and active area of research in my lab, where we are trying to understand that if you have changes in temperature or drought conditions, does that change what kind of microbes are associating with a host plant? And whether that shifts, we use experiments to understand whether the microbes that are joining together with the host in different conditions of light or water, or abiotic conditions, that is, also change how 2 plants might interact with each other given these situations. And would that explain why some species would do better in some conditions as opposed to other conditions? And more recently, we have had some exciting new developments in the lab, where a postdoc who joined the lab has been taking forward some of our work to look at Himalayan ecosystems. And his interest is to look at phenology and how alpine plants, herbaceous communities, respond in terms of their phenology to the changing climate that we see. And this work, we are looking at various angles. We are trying to see whether there are phenological differences to climate between native species and non-native species, and whether species are moving upwards or tracking their phenology along with the climate. And Mushtaqim, who is leading this work and whose idea this is, has done some incredible data collection where he's been tracking the phenology of species, or he has data going back to 2014, I think. And over these 10 years, he is noticing that there is a shift in the timing of when flowers are made by different species, how long the flowers are kept, and what is, when the flowering time is ending. All these phenological features that are critical, because flowering is critical for these alpine plants to attract pollinators and make the seeds that reproduce and produce, maintain the population for the next generation to continue. And very quickly, the figure on the right tells you that among species that are shown in each row of this figure, and each panel is a particular phenological event, which we don't need to go into the details of, but very quickly we see that for many species, flowering is happening earlier, which is what these red lines here tell us, that compared to 2014, they are now flowering much earlier, or they're flowering much not flowering as long as before, but there are also other species that are shifting in the other direction. So some of the work that we want to take forward, which Mr. Kim is very interested in, is to understand which kinds of species are showing what kind of changes when we are looking at these elevated temperatures across an when we're looking at rising temperatures across an elevation gradient. Now, of course, zooming out again, why are we interested in all of this, right? A major reason for why people got in, why scientists have been studying this and looking into the impacts of biodiversity loss is because there was a growing realization that biodiversity is essential or important to maintain certain functions of the ecosystems which might actually be necessary for humans as services. And immediately we can think of pollination, which is a critical service, and we can think of carbon storage, carbon sequestration, and particularly maintenance of carbon stocks in the soil, which are functions that humans need in order for agricultural purposes, but also to stem climate change and the impacts of global warming as we are seeing today, right? Now, this is a very attractive idea. And initial data from many experiments indeed showed that as you have more and more species, you also have an improvement. You have the community being more stable and being more functional or more productive. But when you break down some of this information as more and more data gathers, there are also indications that biodiversity may not be as important for function as we initially thought. So in this example of pollinators, these researchers did what is called a meta-analysis, where they compiled studies, or a multitude of studies from all over the globe, and they looked at what the overall patterns are across all of these studies. And what they tried to see is if you have, or what proportion of potential pollinators actually come to crops, and what is their relative contribution to the pollination service that is happening. And on the y-axis here in blue, you see the number of species, total number of species of pollinators that have been studied or that have been found on these different crops, right? And on this side, on the right side, you see what is the total number of species that is there in the landscape or the study area as a whole. And what we find, if you look at this line, this dark blue line, solid line that is going, these are the number of species that are dominantly visiting crops, and which are very much fewer than the total number of species that tend to visit crops across, as you add up the number of species across these studies. But also, even this line is drastically less compared to the overall species richness of potential pollinators that you find in these studies across landscapes, which shows us that there's this huge gap. You have all of this diversity, this biodiversity, but only a handful or a part of it is actually important for pollination. So we might well argue that for pollination services to be maintained, you don't really need all of this other biodiversity. But that's a matter of perspective, right? So what about all of this biodiversity that is not useful? Do we not study it? Do we not need to know what is going on with it? These insects or these pollinators, for example, might be extremely critical for other plant species that are not important for us, or that plant species which might be eventually important for carbon storage or another service. So indicating that there are all of looking at ecosystems from a human-centric perspective, or trying to understand the impact of global change or climate change on ecosystems from a human-centric perspective, may not give us I would say, maybe excuse or the reason to conserve it, because there is just so much complexity and so much data that we need to understand these hierarchical interactions amongst us. But eventually, regardless of all the science, and even given all of the science, how much biodiversity loss we are ready to accept is going to be a matter of societal choice. And that is something that we need to be deciding on, and what we decide on is going to determine the future or the trajectory of biodiversity, regardless, I repeat, of the science that we do. Because we can go on with business as usual and allow biodiversity declines to take its course, or have some conservation efforts that make a change to what kind of biodiversity we see? Perhaps this is related to ecosystem functioning or something that is useful to humans. Or can we think of a planet where we are preserving biodiversity in all its glory for what it's worth, just because it's just so great to have them around? I don't know where we are going to go, but we do have options, but we are really hanging very close to the point of transition where we need to make our decisions soon, because the legacy of this decision is going to be felt for quite some time. And these ethical concerns, of course, revolve around the fact that we have had mass extinctions before. Things have changed before. Life on the planet has come very close, perilously close to extinction 5 times before, but we are living through a 6th one. And this time it's not because of, I don't know, misbehaving volcanoes or giant asteroids hitting the planet. This time it's because of one species, and a species that understands the consequences of its action. So what might be the ethical implications of that is something we need to think about quite considerably. And I don't think it's science that really tells us that, but it's something we need to think about as a society, because we are leaving a huge footprint on the earth, and most of the footprint is because of how we are using the earth. And in this graphic, for example, is also embedded another deeper problem, which is that this footprint is inequitably distributed among humans themselves, among places of the planet. So what kind of decisions we take and who takes those decisions and who is affected by those decisions is also going to be a major factor that allows us or will shape what kind of biodiversity we preserve in the future of Earth. And something that I think is so much harder than all of the cool science that we can do to understand what's happening with the biodiversity. But this is, I think, a problem that we urgently need to think about and address if we want to think of biodiversity or saving biodiversity for the next generation. And I will leave you with this picture, which I found when I was preparing or when I was looking into material for this talk, which the artist entitles Biodiversity of the Future. Now, I don't claim to understand everything about the picture, and I don't claim to understand what the artist was thinking, but there was something, a sense of urgency and angst and chaos about this picture that struck me as very, as depicting the situation that we are in today and the decision that we might have to make for biodiversity. So I'll leave you with that, and I'm happy to take any questions now.
Thank you,
And sorry, just a last, I didn't speak about a lot of my work, but if you're interested, we are the Community and Functional Ecology Lab, and please do check us out. And there you can see some of the actual research that we are doing, if you would be interested. Thank you so much.
Thank you, Meghna. Thank you for your eloquent talk. It was really, really interesting and gave us such a wide-ranging perspective on what all can happen in the, it's happening right now due to climate change, and where we are going with biodiversity and, community resilience right now. So
thank you,
let's go ahead with your questions. Any questions from the audience? I have one question, Meghna. So can okay, Imrose has raised his hand, so he's on the move. So Imrose, go ahead, please.
Hi, Meghna. This is a fascinating talk. Hey, so I'm really intrigued by the figure that you showed from this, where they did the meta-analysis.
Mm-hmm.
So I think the figure claims that the dominant species,
mm-hmm.
Their abundance is not responding to the presence of other species, right? Is that what I should interpret? It doesn't matter, what is the community diversity is, but dominant species, they remain as the way they are. So I'm just wondering, since this is, I think I'm assuming that this is a more of a, looking at or maybe some sort of catalog in the biodiversity, but has there been any study where they have tried to see the pollination potential of this dominant species? Has there been any functional, studies?
Yeah, absolutely. And I'm not an expert in this domain. This is just something that I put here and I read out of interest. But I know at least there are definitely there is a group in Rutgers University, Rachel Winfrey, who has looked at the pollination, like the pollination success itself. And she has actually mapped the extent of pollination success to the number of species that this can be attributed to. And they've done some very cool work on that regard. And I think what they came up with was similar to the Pareto principle, that only about 20% of the species at most are driving 80% of the actual pollination success, not just visitation. So that's a great question. And for sure, people are thinking of it and they have looked into it. And I think the patterns stay kind of the same.
That tells us that, there's not a lot of redundancy in some of these ecological functions, right? So that makes the communities more vulnerable, in fact. I
guess, that's one way of looking at it, that, if there are just these few species that are doing all of this pollination, does that mean that, if something like a disease that comes? So this is what I mean, like there are different aspects to this problem, right? Like you can say that I don't need 200 bee species or 200 insect species because only 5 of them are pollinating the plant. But we don't know what happens then if these 5 are hit because of something. Do you have other species that will come in and are more diverse communities likely to respond better or be more resilient to these changes and shifts in the abundance of species? I think those are more, long-term studies that, again, I am obviously not the one, the expert to speak on this. I put it here just as a matter of thinking about this. But this is a big question. Like, what do we need the species for? Do we look at it as an immediate use for us or some potential future use? And resilience of communities is a big factor that we need to be thinking about. And it's extremely hard to study because many of these things in most systems we can't manipulate. We don't know what resilience really means until, it is hit and we see that it is not resilient.
I had another question, Meghna. Through all your work, have you noticed some pattern that comes out at you in terms of what happens to biodiversity because of climate change? Is it always impoverishment of communities, or do you think in places like alpine zones there can be an enrichment of sorts, and how does it help or not help the community?
Yeah, that's obviously, that's a great question and something that people are trying to understand. What the global, again, this is not something I have directly looked at, but just a literature that I'm sort of familiar with in the context of the work that I'm looking at. Of course, there is likely to be a zone where species richness is, right? And as a community ecologist, we know that we can just have an increase in species richness, but we can have a major shift in how abundances are distributed among those, right? But you can have these zones of overlap, right? Where particularly like with species, for example, moving up the mountain, there are certain mountain zones where you might have species displaced, but maybe central areas in the mountain where you have a sudden increase in species richness because now you can have things moving up or coming down, right? At larger scales, going up the latitude, you might have higher species richness in some of these mid-latitude areas because now they can support species from both, above and below. And some of the warm area species might actually be going there and be able to live in the higher latitude. So of course it's going to happen. And I'm very sure people are looking at it. There are these databases that people are using to look at it, like Biotime, for example, is a database that people use to look at some of these changes in species richness. And I think they're seeing these kind of idiosyncratic responses also. But by and large, we are likely to lose species at a global scale from many areas. And the most proximate driver of that, honestly, is probably going to be land use change more than before the climate. The climate is going to become more and more important for sure as things get warmer, but it's going to be like a double whammy.
Thanks, Meghna. That really was illuminating. If there are no more questions, I think we've seen a lot of stuff. I think it's tough to absorb everything so easily because there's just so much information in here. But amazing talks. Thank you all for coming with your talks and presenting it. And I'd like to just thank all of the people who've been involved in setting this whole conference up. I can see that Gautam is no longer here, but he's really been instrumental in kind of pushing us to do these seminars and kind of holding it together. Astha from 3CS, who's really been handling everything since day one. Imrose, of course, and Ian, we're very grateful to you for joining us today from really far off. Yes. Thank you all. And we look forward to having more exchanges like this and also following up with each of you on your amazing research and all your results. Thank you.
Thanks very much.
Thank you. Thanks, everyone. It was
Thank you.
fascinating.
Bye-bye.
Thank you.
Bye.
Bye. Thanks
Bye.
for inviting.
Bye.